We're just stabilising the ROV in the water column so we can get a better view of that siphonophore.
It's often easier to identify a lot of these gelatinous animals by looking at video rather than still photographs, just because there are behavioral cues such as that spiraling by scalia that can help you tell the animals apart. There we go. So in these very shallow waters, it's quite challenging to pilot an ROV because the ROV is by necessity attached to the ship by its umbilical cable. The SuBastian's umbilical cable has floats attached to it close to the ROV to help keep the cable away from the ROV and help keep it neutrally buoyant. So we can do a little bit more floating rather than like hard power with the thruster to try and keep our position in the water. But the length of the ROV is constrained in shallow waters. The length of the cable, sorry, is constrained in shallow waters. So we're at 210 meters depth. I don't know if you have a readout on how much cable is currently being paid out there.
To 83 meters, so there's a lot of slack in the cable so there's not jerking between the ROV and the water column and the ship above.
That's right. So the deeper we go, the more play we normally have in the cable to allow us to do longer observations on each animal. Usually in the shallow waters, you get to spend less time for each observation because you run out of cable faster than when you're deep. So I would imagine that would be the same here with the SuBastian. Okay, so we're slowly proceeding through the water, looking for gelatinous targets, or actually any targets that we see to help us understand more about the midwater ecosystem here. The ROV has all of its lights on. In shallow waters, a certain amount of light reaches depths, the ambient light from the sun, especially when the water is very clear. This makes it quite hard to see the animals because they tend to fade into the background. As we go deeper, it gets easier and easier to see the animals because the contrast is higher compared to the background. I would say at the depths we are here, we should be able to be seeing the animals at a nice contrast. So if there were animals here, we would be seeing them. What's the water temperature currently, Rob?
18.5 degrees, so it's definitely cooled. But you know, these waters are very clear. If we go over to the adjacent coral reef, which is only about 3 or 4 kilometres from us, we can get calcareous coralline algae down to around about 200 metres, so just a little bit shallower than what we are now. So that's about 0.5% of the light that you'd find at the surface. So these waters are typically very, very clear here.
So for the animals that are adapted to the twilight zone, if you've got enough light to photosynthesize, for them it would be like entering a room with all of the floodlights and spotlights on and suddenly being blinded as you're there, because these animals can detect light at much, much lower levels than the levels needed for photosynthesis. So we're still in brightly lit surface waters if you were a mesopelagic fish or some shrimp with eyes. Large mucoidal particles coming into view. These strings can also be, well, we call them strings when they're big, long, elongated particles. They can be produced by a variety of organisms as well. Sometimes it's just a more rounded particle, which as it falls through the water, slowly becomes more and more string-like in shape, because the surface, the cross-sectional area becomes smaller and smaller, and the particle kind of wraps around itself and becomes more linear as it's sinking. So you normally find more of those linear marine snow particles the deeper you go, and the more rounded, fluffy-looking ones are closer to the surface. So when we see these string-like particles near the surface, there's a good chance that they were produced by radiolarians or other rhizarian single-celled plankton, which tend to produce mucus strings, hang them down in the water column below them. All sorts of animals are attracted to these mucoidal structures because they think they're good food, or the bacteria and other things will grow on them. And then this single-celled protist will haul in its mucus and eat it all, and get the energy back from the mucus itself, but also from all the animals that are attached to it. What do we have here? Looks to be more mucus. Oh, no, it's a siphonophore. That could have had a bell there on the top, looked to be hanging down and then across in a fishing position. So I'll have another look at that with the high quality video later to see whether that was a small calicophoran siphonophore or not. Normally in these upper layers, you find many siphonophores. That's those colonies of jellyfish polyps. And the calicopherans are very abundant. These are animals with no float. Instead, they have only swimming bells. Oh, this looks like a siphonophore. And it might be a case of speak to the devil. It looks like it could be a calicophoran. The white spots occur quite a long way from each other, which would suggest it's maybe a prayed calicophoran siphonophore, which you can only tell really by looking at the nectophores, the two swimming bells. Calicopherans can have a variety of different types of swimming bells. One of the abundant types has a rocket ship shape, where the upper bell is a triangular prism and the lower bell is a rectangular prism, both of which can expel water. And this streamlined shape allows it to, when it escapes, to move rapidly through the water. The other type is the prayed siphonophores, which have two rounded or cylindrical nectophores. So they move a lot slower, but usually the prayeds have much, much longer tails or siphosomes, many, many more stomachs. So they tend to have big, bulky, rounded nectophores that are positively buoyant. They change the concentrations of ions within the jelly so that they have lots of light ions compared to the ion composition of the seawater around them, which helps the jelly to float. Light ions, and so when they need to swim, they can swim with the muscular nectophores, the jelly, but otherwise they just kind of float there and hang around. So when they need to swim, to jump into their tentacles. These sit and wait kind of predators normally eat larger organisms as food, because when you're just sitting there and waiting for the food to come, the food that tends to come into your tentacles is fairly large, and they swim or they flick onto your body, which is where you consume them. Other animals tend to swim through the water and disturb animals and make them flick onto their tentacles by doing that. So some animals will sit and wait. Some of them will swim and use that swimming motion to collect the animals on their tentacles or on their lobes in the case of comb jellies. (232 meters depth). So far, most of the animals seem to be small calicophoran siphonophores, except for that big scalia Siphonophore colony. Some of the mucus structures may be the tails or cyphosomes of siphonophores, where they've been disturbed. And when they escape, that rapid propulsion by the bells is enough to actually cause the tail to detach or dissociate. And the tail will go on living because the zooids there can feed by themselves, they can reproduce by themselves. The only thing they can't do is escape or swim. And so these tails will gradually sink, eating and reproducing as they do, until they run out of energy and they die. And as they're degraded by bacteria, they look more and more like mucus. And in the end, they're just a massive tissue that's left over that the bacteria can't digest. And other animals come along and chomp on them as they sink down towards the seafloor, providing food.
Dougal, ultimately, it's a huge amount of material that ultimately ends up on the seafloor, either as, larger dead animal carcasses, and I'm just thinking of, copepods, I'm not talking about whales and the like, but all of this mucus. Have you done much studying about what happens to all this mucus at the seafloor interface?
There are very few snot scientists. Oh, yes, mucus scientists around. A lot of work has been done with fecal pellets, for example. There are people who can look in sediment traps and look at all the different fecal pellets and see if it's from a copepod or a krill or something like that. Much less has been done on the mucus. I mostly work with jellyfish. So these are very fragile, gelatinous animals, which are hard to survey and hard to sample in themselves. They get broken up in plankton nets. So we suck them up in suction samplers or slurp guns with ROVs such as this. Ooh, what do we have here? Mucus is even more fragile. So it's such a challenge to research. There's been very, very little work done on it. Ooh, wow. Let's get in nice and close for a look at this.
Yeah, we're just positioning the ROV so we can stabilise it and get it still in front of us.
It looks a bit like it might be a pyrosome, but it may have something attached to it. The way it's moving, you can see it's pretty small.
Yeah, if it gets too close, it drops out of the light, so we're just trying to reposition here.
Roger that. Yeah, so this mucus...
May have lost it there, Dougal. No, I didn't. It just looked like, just as you described, one of those detached series of bells. There didn't seem to be any other structure on it.
If we find one of those with the nectophore still on it, we should try and sample it, because there's been a few of them. So it would be nice to get a positive ID. I'm not saying it wouldn't be possible to do it with this.
We're going to try and capture this one.
The bracts for Pred siphonophores are definitive, so I could get a species ID from this cipher zone. It looks like it's time to do some ramen noodle slurping. These repeating units, each of those white spots you see is the stomach of the animal, because it's the most reflective part. It's a set of repeating units. Each one is called a chlamydia, and it contains a stomach, some kind of reproductive zooid. Looking for prey and have more of a chance of getting at least some food. So
Yeah, we've got that on board for you, Dougal.
awesome.
Nice.
You go take a look at it in the canister.
Yeah, we can swing around and do that. 255 metres. Have a quick look, yeah. Thank you.
Hi, this is just a test for you guys, so we can spell Siphonophore. You know how to spell it?
Predation.
That's canister one, isn't it? Where he is?
Using canister 1.
Is that on the back side there?
It's very small and transparent.
Very hard to see. Yeah.
Okay.
We can't make it out here.
Fingers crossed.
Yep. We'll confirm later on.
Sometimes you get stuck up on the grading there, but
And so in the ship laboratory now, my colleagues there will be extracting that animal from this Is that an alciopid worm or a tomopterid or something spiraling and swimming there? In center screen, up near the top. Way to go. Ah, it might be really close and out of the light. Or like the whole animal rather than just the parts is really invaluable for doing this kind of survey work. The same can be said for seafloor surveys. When you're zoomed in on a sea star, for example, a wide angle, fixed focus, a fixed field of view camera can give you We're below 250 meters now. What's the water temperature, please?
Currently 16.9 Celsius, Dougal, at 250 metres. 1 to 6.9.
Are our oxygen levels also greatly decreased from the levels at the surface?
We're not tracking that live, Dougal, but
Let's do that.
one moment, or does another screen's come up?
Oh, another for scalia?
Yeah, Dougal, the sensors on board this SuBastian are typical temperature, salinity, conductivity. We measure sound velocity as well. That helps us because that's all about using sonar, pressure, depth, and oxygen. So they're the main sensors on board, and all that's logged in real time as part of our every single dive.
Weird. Possibly not, no books. Really, it's swimming like a jellyfish. Oh, it is a jellyfish. Is that a jellyfish? It looks like a Pandea.
Yeah, it's pretty active. We're just trying to stabilise the ROV, and then if it's too close to the camera, it gives us problems too.
And getting a species ID, when we catch a pteropod shell that has polyps on it, we can then DNA barcode them and see if we can find a match. Because these polyps don't survive without the pteropod being alive, and so you're unable to rear them to the point when they are mature enough to tell what species they are of an adult jellyfish. So DNA barcoding is allowing us to determine the juvenile stage and the adult stage and see if they're the same species or not. Which in surface animals, you're often able to keep them in aquaria or something and do these rearing, husbandry kinds of experiments. But with deep sea animals, normally they don't survive very well under captivity. We keep our eyes out to see if we can find another.
Yeah, I think we lost it there, Dougal. Sorry about that.
Thanks for that one, Mitsko. What else can we see? Getting down towards krill territory soon. Having a lot of krill around will increase the number of predators as well. Oh, what's that? Fish? We should all.
Left or right, Dougal? On the left, thanks. It's got lots of spines on it. It kind of reminds me of those juvenile crustaceans, the shapes they form. Yeah, definitely. Looks like a little crayfish, juvenile crayfish or crab. Got very obvious eyes on it. It's got things like a mosquito.
We know so little about larvae that if it looks like we're able to suck him up, I think we probably should. Knowing that they're free living rather than attached to something and the exact depth they are. For the larval stages can help us to see what the effect of global warming and increased water temperatures might be. Because if they can't handle the heat and they can't be close enough to food, they're not close enough to the surface, then the species itself is in dire straits. Any info we can get on the larvae is good info to have. But to get an ID, because the larvae, all the different stages probably aren't morphologically laid out yet, we'll need to catch him and get a DNA barcode to see which adult it matches up to.
Yeah, a colleague of mine, Teresa Corrett, was doing light trap work out here at Osprey Reef, mainly looking for Irukandji jellyfish, but these are sorts of things that get attracted into those light traps as well.
Right.
Slurped up.
Oh,
Keep an eye out that we actually have him in the canister, because he may swim back out into the hose and get sucked into the next canister during our sampling of the next animal.
roger that.
Very nice. Keep on heading down. I'm going to try and make 1,000 meters today so that we can compare data sets with other areas we've been surveying around the globe.
Allow us to get data of the distribution of animals
In different ways, depending on the oceanic area we are surveying. This will help us to tease apart the factors that are most important in determining the distributions of the animals. Are they temperature dependent? Is it more oxygen? Is it salinity? Is it light levels? Etc., etc., and how important each of these is in determining their habitats. Coming down to 280 meters depth. In Japan, this would be where we would find krill. Probably here, because the transparency is so much higher, we'll find them a bit deeper. And I wonder about preyed siphonophores, because this would also be the depth at which we find Preya dubia, one of the longest planktonic animals in the world, 40 meters length documented. They kind of hang up above the krill layer, waiting for the krill to pass through on their way up to the surface, and hit them with hundreds, if not thousands, of stomachs with tentacles. And then when they're on their way back down in the early hours of the morning, they hit them again. And if we find a preya, we'll know if they inhabit the same depth, regardless of whether euphausids are. Oh, big jelly. Have you saw misses?
Looks like a Siphonophore. Same one. Just altered the position so we get different light angle.
Yeah, so this animal was first described by scuba divers in surface waters, and the jury is still out as to whether this animal with the developed gonads here is actually a different species or the same species. We're going to have to end up getting DNA barcodes, I think, to be able to tell that, because it's very hard to rear these animals. They're 98% water and very fragile. Thank you for that. Siphonophores are also notoriously difficult to get sequences for. There's all sorts of weird stuff happening with their genes. They're a very ancient phylum. And there's a lot of discussion in the literature as to whether they're older than sponges or not. But they're right up there with sponges and cnidarians at the base of the evolutionary tree, early branches.
Dougal, is there any fossil history of them? You know, any like imprints in sediments from fossil record?
That's a very controversial question there. There are animals that have been described to be fossil ctenophores. Because there's a lot of convergent evolution in morphology that happens as well, it's very hard to actually say they're ctenophores. The ones I'm aware of now are benthic, and they have more than 8 comb rows, for example. So we have benthic ctenophores now, so that's possible, but nothing that has more than 8 comb rows in any of the extant taxa. So whether they are actually ctenophores or whether the comb plates of ctenophores have turned up in other lineages, we still do not know. The bodies are very soft, even softer than cnidarians in most cases. So apart from the comb plates, there's normally not very much left to identify them as ctenophores. They also seem to have gone through a bottleneck with regards to extinction events. So the current diversity in the phylum seems to have radiated out from only a few taxa that survived a mass extinction in the past. So genetically, they are very close to each other, and very small differences in sequences, possibly due to gene rearrangements, perhaps, have caused a big diversity in body forms in all the extant species. So it's very hard to look back in time to see who's related to who because of that bottleneck that they passed through, making all of the genes fairly related in terms of sequence. That was pretty geeky, wasn't it?
No, not at all. It's ship's half filled with marine geologists, so quite interested to hear this. I guess extinction events, you're probably talking about the last big Cretaceous tertiary extinction event.
Well, without a fossil record. It's hard to tell which one.
Yeah, and I think of Australia, which is a very, very old continent, and there's some quite old sediments there. And I think of the Ediacaran fauna. Oh, there's a jelly up in the top right. I'll shut up.
Nakamedusa, top left. Tag him and bag him. Let's get some good video of him. This is one of our main target species. It looks like it's got an amphipod maybe on it. Or we might have eaten lunch. It looks to be on the external surface of the bell. solmices to catch a jellyfish, and then he'll pop up there and steal the food from the solmices, and he'll eat the jellyfish. So we may see, if we get a good bit of video here, that the tentacles may have been eaten by the amphipod. There may be some long ones and some short ones.
So you'd say this amphipod is parasitic?
Yeah, I believe we'd call it parasitic.
Just working on the focus here.
Gee, it's a fast swimmer. That's nice. You see the tentacles are held in front of the bell. This is so as it swims through the water, its tentacles come before the bow wave that it's producing as the umbrella is moving through the water. So it can cause the tentacles to collide with its prey before the prey knows that it's coming. Okay, so we attempt a sampling of this guy. Doesn't look like he wants to show us himself looking from the top or the bottom. He's just trying to swim and get away.
Yeah, it's pretty active. We're going to try and sloop him up if we can.
Okay. Oh, there we go. You guys still have a visual in one of the other cameras?
Yeah, we do. The sit cam's a bit wider, we can see it.
Great. I see it. Come on, work with me, solmices. Very small.
slurp him up.
Awesome. Bag bags. Great. So hopefully we'll be able to look at the morphology of this, and we believe there are several cryptic species or hidden species that are now currently referred to by the same name, which are actually different species. And by getting a pristine sample and a DNA barcode as well to go with it, we should be able to tease that apart. The mucus particle, maybe like the fluid libation. My screen's a little dark here for seeing things at present.
Siphonophore coming into.
Looks like the Nectaphore is Nectaphores to the right, middle of the animal there. It's coiled around. What do we have? Maybe a Euphraid? Zoom in on those swimming bells and let's see what we've got. This maybe that rosacea that we were looking at a little bit earlier. To tell the genus, we need to look at the swimming bells and see there's a very white reflective structure inside them called the somatocyst, which can be bulbous, can be branched, it can run along the surface of the bell rather than penetrate the mesocelia. And by looking at the shape of that structure, which is used to store food and nutrients, we should be able to tell what genus this Siphonophore is in. Wow, look at those tentacles. This also looks like it might have something attached to it. And this is the cyphosome we're looking at, all the chlamydia that are attached to each other in a repetitive structure. The other end of the animal is where the swimming bells are. This is also fairly small, isn't it?
Yes, we think so, Dougal. We're really zoomed in on this guy. Just playing with the focus, I am manually trying to.
So when we collect him, we'll need to try and suck him up from the Nectaphore end. This end we're looking at right now.
The end we're looking at, yeah, we can try that.
Grab it from that end rather than the other.
Yeah, bottom up. Let's try that.
Yeah, copy that.
So this is a
there,
relative.
Dougal. Yeah, we just might have to maneuver around, maneuver the ROV around to the Nectaphore end.
Okay. Yeah, I have a feeling if you did suck it up from the siphosomal end, the mesh inside the canisters would probably get clogged before you manage to collect the nectaphores. Let's do the spaghetti slurp. The really difficult thing about collecting jellyfish in midwater is when you try and reverse the ROV, there's often water propelled by the thrusters out, which changes the position of the jellyfish and can blow it out. So, whoa, there we go.
We got him.
So it's quite a challenge to try and keep these little gelatinous animals out of the wash of the thrusters. With the slug. Great. So
Yeah,
that's also another Preyed.
Yeah, confirming we've got him in the canister there, Dougal.
Excellent. So this is probably in the same kind of niche as the Preyeds off Japan that hang around above the krill, because we've seen a few of these and not so many krill yet. I would imagine go a little bit deeper, we'll start to see the krill kick in. Lightweight predators.
So, Dougal, we've probably got an hour and a half at most on our day pelagic dives to left.
Okay, let's keep getting going.
Yep.
No, sorry, that was our 2.
Oh, right, shot two.
Yeah, sorry,
Now we're on,
we did a little backwards today.
Was it?
So we got Chris and Adam, our ROV pilots. They're taking a full team here. We're taking turn about every few hours because there's quite a lot of concentration required to manoeuvre SuBastian. We've got some ROV pilots with us on board, and real pleasure to be in the control room with them.
Looks to be another rosacea, another Preyed siphonophore. There's a few of them around. Just get good enough video to see whether it's the same thing. So the chances are that is. Do a quick zoom on the nectaphore if possible, and then we'll keep going. Yeah, I can't see a globular somatocist, so it does not look to be desmophies. It seems like cylindrical nectaphores, so it's probably a rosacea. And we have caught one, so we should be able to tell. Oh, wait a minute. Are there globular somatocysts in there? thought I just saw reflections on either side of the stem up in the nectaphore there. Yes, okay, so this is Desmophis annectans. That's a species ID for that guy. So thank you.
Do you want to click
I wonder
that?
if that last one is the same thing. No, I've got the ID, and hopefully the last guy will be the same thing.
Roger that. We'll move on. We'll move on.
Thank you.
Yep, thank you.
Desmophis, Desmophys, annectans, Annectans.
Dougal, got a question here. Can these siphonophores sense light? Are they disturbed by the ROV lights?
I can't provide a definitive answer to that, but I have done some experiments with, oh, wow, what's that? With some midwater medusae, hydrozoans, where I've shone light on them and they stop swimming, take the light off and they start swimming again. And so although they don't have eyes, they're able to directly sense the light, probably through their nerves or possibly from epidermis. So I'd say the chances that the siphonophores can detect light are high. But we have no experimental evidence to support that. Coming up on what looks to be another Forscalia, perhaps? Yes, and that looks to be Forscalia formosa, I would say. Yes, Forscalia formosa. Thank you. The last Forscalia species we had, the nectosome was very short and was flattened at the top and kind of square looking. This guy has a tapering nectosome. Also the scyphosome here, the Christmas tree lights that are wrapped around the body, here are also tapering down here in Formosa, whereas Edwardsi, which we saw a little bit shallower, had a very, very long scyphosome, and it didn't taper. It was almost the same width right down to towards the end of the scyphosome. So these two Forscalia species, it looks like Edwardsi lives shallower and Formosa is living deeper. So they're partitioning up the habitat based on maybe depth, maybe temperature. We're not sure yet until we get a variety of these different physicochemical parameters and distribution data on the different species. And because the nectaphores are arranged in a helix rather than biserially, as it swims, it spins through the water, which is diagnostic of the genus Forscalia and the family Forscalidae. Thank you for that. Let's keep heading down, try and make sure we get to 1,000 meters. Still no krill. Of course, there may have been krill while I was getting so excited with the Desmophis and the Forscalia, and we'll know that when we look through the high-quality video later, frame by frame, analyzing the video record taken by the ROV. If anyone sees a krill, just chime up. The environment these animals live in determines where they are distributed. But it's not just the physical chemical environment which is important. It's also important if their prey is there, and also if the predators are not there. So another jelly on the top of the screen. So we're interested in seeing what kind of prey is occurring in the animals as well. Coming up from 330 meter depth. Off Japan, the highest diversity is found between 400 to 600 meters usually. This is probably because all the diametrical migrants that head up the surface to eat at night time are all coming down to around those depths during the day. The jellyfish diversity is also high at those depths, and we don't yet know whether that's because the jellies also migrate, and the distributions during the daytime overlap with the animals that don't migrate, or whether it's just the high diversity of prey that occurs there means there's a high diversity of predators. We have here. Looks like ooh, is that a heterobranch? Oh yes. The far one is marine snow, but the front one looks to be a mollusk. A heterobranch mollusk. We call it the elephant jellyfish in Japan, but it's actually look at that. It's a very long filament coming off its tail. The top right white spot is its jaw, its buccal organ. And there he goes, swimming away. And that longish cylindrical thing in the middle is, I think that's probably his digestive organ. There should be a shell probably attached around that area as well. Although it may be embedded in the jelly. Very nice. And it's only got one fin on the top of the animal there, whereas other pteropods have two, which they use to swim around. You can also see the eyes there behind the mouth, quite a long way behind what looks like an elephant's trunk, which is the mouth of the animal. I believe these two prey on gelatinous organisms. I guess when your body's transparent, you want to eat something else that's transparent as well, so that your food can't be seen by predators who might be looking for something to eat. Cool. He's looking back at us. He's doing backstroke there. His scientific name escapes me right now. I should know it. Fio, F-I-O. That'll be a job when we get the video and are doing the analysis later. Very nice video. I wonder if it's got any jellyfish parasites living inside its body cavity. Can't see any. Okay, very nice.
Does this need sampling, Dougal?
No, that should be okay. We should be able to get an ID with that.
Okay, onwards and downwards.
Yep. It's transparent and jelly-like, but the jelly is a little bit harder than regular jellyfish, so they have been caught in nets, and there are specimens probably from off Australia as well. So we should be able to look at the morphology and get an idea of that. It helps that they have hard parts like jaws and shells. And they swim pretty fast if you actually try and sample them. They just suddenly, woo!
So we just swapped out the ROV pilots. We've got J-Rod and Cody on controls now.
Roger that. Coming up on 350 meters soon.
Temperature 12.8 degrees C.
Roger that. Mucus. Sort of part of a discarded larvation house, perhaps. Is that a siphonophore? No, it's just a mucus. Looks like it's mucus that may have been a siphonophore once.
Siphonophore in front of us.
Yes. The Chlamydia seem to be less reflective and large as that Desmophes we looked at just before. Is that because it's nectaphores and it's a ghost tail it's fishing?
Yeah, it could be. I don't see any. Oh, it looks like we've got Ctenophore just drifting across the screen at the moment.
Oh, let's take a look at him. Is that very long and belt-shaped one, or looks like a mushroom?
Yeah, very long. Flat.
Very long ribbon shape. Yeah, that's cestum. Cestum veneris. Cestum. Venus's girdle. You can tell from the canals branching out from the stomach, whether they branch out directly from the stomach or whether they're curved from the arboreal organ, exactly which genus it is. These animals can be quite long, over a meter in length. And they swim around sinusoidally like a snake or an eel larva swimming through the water. They swim like a plane, so rather than when they escape, they tend to swim like this, but they can also swim like a hydrofoil going through the water. Quite interesting animals. They have evolved such that to their detriment, that if a scuba diver pokes one, it rolls itself up into a little ball, which makes it easier to put in a jar and sample. I bet they wouldn't be expecting that when they were evolving. Thanks for that. I'll keep on heading down. See what else we find. It's a fairly deep record for a system. Something else? No, that's something different. Get rid of that. Nichol in Japan is saying that's a Pterotrachidae, that tetrapod mollusk we saw just before. Siphonophore perhaps?
Yeah, quite small. It's swimming.
Oh, looks like a python in there, maybe? Does it have multiple bells on the cytosome? On the next sternum, I mean? Okay. We should keep going down, I think. Really like to make 1,000 meters if we can.
Yeah, it's about 1,300 metres to the seafloor where we are. So after your dive finishes, Dhugal, we'll continue down and be looking around that sort of 13 to 1,500 metres depth on the seafloor.
What's the shallowest you expect to be coming to at the end of your dive?
Probably about 1,200 metres. We're 4 kilometres from the edge of the of Osprey Reef on a very large block.
Roger that. Oh. This looks to be the barrel of a front amphipod, perhaps, but I can't see the amphipod inside. Or maybe it's the pyrosome. Looks like the zooids are still intact, but it may just be a pyrosome. But it looks like it's open at both ends, which is not normal for a pyrosome. We can get an in-focus shot of it. There we go. Yeah, so it looks like it's the barrel of a front amphipod, and the amphipod's been eaten or has vacated it. Thank you very much. So these barrels are made from, usually from tunicates, sometimes from siphonophores, and an amphipod, which is a little shrimp-like crustacean, catches the tunicate and scrapes away at the outer surface and fashions itself a barrel in which to lay its babies. And it looks like this is a barrel that had been made from a pyrosome, which has been abandoned, either because the front has been eaten by something else or for some other reason. Mucus.
Yeah, we saw a live one yesterday, didn't we? That was pretty spectacular. The pheronemer inside.
Yeah. So people can look back at the video from yesterday and see the female, the mother pheronemer inside one of these barrels. Some pretty awesome video there, looking down directly on its head, for example, into its eyes. And that was also in a pyrosome-based, biology-based barrel. Something else coming up mid-screen here. What's that? Going off the bottom of the screen. We have something else coming in the top. That looks like another for scalia. Yes. I think it might be for scalia symmetrica, but I couldn't be sure. Oh, wait a minute, is that a Siphonophore? Does it have very long palpons, great big long sausage-shaped things coming out from around where there's orange tentacle filaments?
I wouldn't say really long, but it's about the width of that head, and in the tentacles about that same distance out.
Okay, so this could be Siphonophore. Yeah, once it looks like we're getting good video here. If we can suck this guy up, that would be good also. There's very few records of this animal in the literature. It was only recently described by Phil Pugh from Southampton. And I don't think I have a sequence yet.
That one was easy.
Great. Yes. Physophora, Physophora, Gilmeri. I think one of the Physophora species has bracts and the other one doesn't. Can't remember which right now. And I've only got one observation of that off Japan right now, where we're certain of the species. So that's great. Similar depth. Interesting. He's in there somewhere.
Yeah, at the bottom there.
Okay. Let's keep going down. But there's an amphipod swimming around there. Wow, I haven't seen that species for over 10 years. Very cool.
Looks like another Siphonophore in front of us. Strange shape though.
Yeah, that looks like another septum.
Oh yeah, the girdle Siphonophore. It is coiling up.
Kind of wonder why they evolved to curl up. I guess they're trying to decrease the volume they're taking up, so they're not making as many vibrations in the water for a predator to find them. But it really does make them a lot easier to eat when they curl up. If you've got a big mouth on you. Okay, once we got some video of that, we'll keep on heading down, see if we can cover the whole water column here. Is that another? Oh, it's a load bait. Looks like a Euramphea, perhaps. Does it have filaments coming out from the arboreal end? Where are you? What is that? No. Oh. If there are no filaments and the body looks, if it's like a V-shaped body, it's very laterally compressed. The body is V-shaped. It will be Kiohimea usagi. And if it's more rounded, it's Kiohimea orata. It's looking a lot like orata to me right now. I don't think I can see any filaments on triangular projections on the aboral end. Yes, so Kiohimea orata. Nice. This is the first record for the southern hemisphere, I believe. Oh, you can see the tentacle bulbs there down by either side of the mouth. It's a Diopia / Kiohimea animal. We're now looking as you would be if you were a prey item and it was swimming towards you, with the lobes on either side and the mouth pointing at you, and the tentacles hanging there to catch you as you try and jump away and escape and run into the lobes which are surrounding you on either side. Very refractile, beautiful rainbow colors there from the comb plates as they reflect the light of the ROV. Yes, it's great. Wowie kazowie. The oracles are actually quite short and stubby, really. Kiohimea usagi, they're much, much longer. Okay, thank you. It's a good video. Good documentation there.
So Dougal's got
We're
back
just
here.
seeing too many interesting animals.
So timekeeper here, we'll give it another hour on this descent.
Roger
If
that.
we can
Down.
get as close to 1,000 as we can.
Ignite. There's a krill. We have krill. More krill. String. Oh, another Siphonophore. Another cestum, or is it an eel larva? Looks like a cestum. Yeah, you can see the waves of color going along the comb row. Wow, so when they're undisturbed, they're in a C shape. That's cool. Let's keep heading on down to make sure we make our 1,000 meters. Some more krill there as well. Yeah, you can see the krill are spinning as they swim. So that's probably the genus Euphausia. And that one's donutting as well. I have a feeling krill can probably see polarized light, and they're spinning around there to get looking at different polarization of the particles above them. about time for another solmesis, I think. Oh, there you go. Speak of the devil. Oh, has he caught something there? Oh no, it's the particle behind it. So once we get some good video of this, if we could slurp him up as well, that would be great. Does that have stomach pouches? That look purplish. It doesn't have very many tentacles. Okay. That's nice. Okay. Let's sample him. 1, 2, 3, 4, 5, 6, 7, 8, 9. Wow. Very few tentacles. Kidding me? Krill? Did you catch him? I think your audio is turned off now. Yep, that looks like a successful capture then. Hopefully he's somewhere in there. Fingers crossed. So we're at 410 meters depth, heading on our way down, hopefully to about 1,000 for our midwater transect. And just checking the suction sampler canisters now to see if we have animals inside, after what we hope was a successful sampling event. Resuming survey. Looks like a salp. Solitary salp zoid. Swimming up to the top of the screen. Marine snow. That looks like it might be a Medusa. Can't tell what that is. Is that the Medusa? Four radial canals, perhaps? Small stomach. Looking at a microchromatograph or something else. It's a good video. It'd be a shame not to sample it because we need to check on the bell rim to see numbers of statocysts, if it has cordiali, etc., in order to get an ID for this. I don't think it'll be possible from the video alone. So if we're able to suck him up as well, that would be wonderful.
Last jelly Dhugal, is that all right?
Yep, that's cool. You went in? Okay, let's head on down.
Hey Dhugal, what do you want us to call this one?
Leptomedusa.
Okay.
Lepto.
I got it. Thanks. Leptomedusa 33.
You have to have close up to the bell rim or look at them under the microscope later to see in order to get any better than that.
Want me to do that?
If you're able to get like a close shot of the bell rim, that'd be great.
Do my best. I'll make a note. I have a USB microscope. Could try to use.
Okay. I know how hard it is.
It's
with a vibrating weapon.
I dread the moment. I dread the moment when I send you my pictures and hear your response.
Well, a blurry out of focus picture of a staticist is better than a staticist dissolves in formalin and it's no longer there.
All right, good to know. Try to get one or two focus shots. Often it's hard with the scale underneath, and they're see-through. So we try our best.
I feel for you. I'm very appreciative.
Oh, no worries. It's a fun challenge.
Once COVID is over, I see pints on me.
Well, you could also return the favor by finding some black corals and going through the painstaking effort of taking images of the skeletal spines, then we're even.
Okay. Bit of mung, marine snow. I find a few octocoral larvae actually in the midwater from time to time.
That's interesting. I know for the black corals, we don't really know much about their reproductive strategies in the deep. Well, we know nothing about their strategies in the deep.
Siphonophore. This looks like Tetraphalia, which is an undescribed genus and species of Siphonophore. Yep, it's got those tentacle bulbs that are angled in an axis other than abor. Just get one shot from the 90-degree angle to this. That should document him okay. These things eat krill as well. Nice. Yep, that's him. Undescribed genus and species of Siphonophore that we currently call Tetraphalia for want of a better name. Thank you. Just keep going down. Octocoral larvae, or like pre-metamorphosed guys, don't have spicules, am I correct?
I don't know. Sure.
Have a survey in December diving inside and outside the volcanic caldera in the Larval Island chain. So if I see any octocorals down there on the seafloor, I'll remember picking one up.
And stick with the hexacorals.
Hexacorals, okay.
Octocorals. Every trip we go on, I find something new, just like you with the jellies, so together we can taxonomically describe them all.
Is this another soul missing? No. No, it's not. What is this? Hmm? Looks to be a Medusa. Could it be dead? It doesn't look to be very happy. Now they ate a porcupine.
Many transects behind.
The Medusa running up the hill.
Work.
Oh.
The polychaetes.
It looks to be dead. So we don't know from what depth it's sunk from. City?
Besides.
Come on, let's get up here. There we go. Bit of Pagantha. Looks to have a photo profile. That was cool. Great to see a live one of them somewhere.
Don't you have students to do this kind of work? Would work. I couldn't even do it.
So many jellies.
Yeah.
Never gets to 1,000 views if you look at all of them. What are you?
They're doing it fast. Sink some stuff. Like,
Yeah, let's keep heading down. Ooh, another saw misses.
There's too much.
We know there are Paganthas now, so we really have to check to make sure there are no other forefathers on any of these animals. Looks like a promises to me. There we go. Okay, once we got the money shot, we'll keep on going. Nice. Oh yeah. Awesome. Certainly a lot of life down here. Is that a Siphonophore fish, bristlемouth perhaps? Is that another prayed Siphonophore? Looks like there's green fluorescent protein there fluorescing back at us. Could it be a Liliopsis? Although the cyphosome doesn't seem to be fluorescent. Yeah, hopefully there'll be enough frames in there to tell what that is. The squid. Yep. A firefly or something else. Very reflective body, the guanine crystals or whatever it is. Oh, it just ejected. Running around. How do you like that? Just spinning around on his axis. So quite an interesting posture for a squid. They're normally more horizontal and looking down to see if predators are coming to eat them. But this one, the arms are hanging down below it. Looks like it's got luminous organs inside there. Possibly on the tips of some of the arms as well. Big digestive gland. Okay, I've got a lot of good angles there. Should be able to identify that, or Mike Vecchioni should be able to at least. They just keep coming. Looks like it might be a Kiahumea, unless there are filaments on the ends of those projections. I don't think I can see. So, Kiahumea usagi. Yes. Okay, that's good. I got an ID for that. We still don't know if the Kiahumea we saw a little bit shallower that I called orator is actually the juvenile stage of usagi, which we just saw now. The jury's still out on that. Orator is much more rounded. Usagi is much more pointy, like a triangle. And it seems the original description of orator was done on a damaged specimen. Oh, there's a siphonophore. So it's very hard to be definitive when you have no holotype and the original description didn't have all the characters you really needed to look at when you find subsequent species. At the time, of course, the characters were enough to tell it apart from any other known species. But unless you're very meticulous in recording all of the morphological characters, you find it's hard to tell new species you find after that first description apart from each other. Here's another Phronima in its barrel, like we saw yesterday. Phronima sedentaria. This one is hanging down there with just its head inside and is spinning around. And it'll try and catch a jellyfish in those claws there, and get the jellyfish tissue and send it down to its babies that are attached to the inner surface of the barrel. This one doesn't seem to have laid any babies yet, so I guess it's fashioned itself a barrel fairly recently and is feeding up in order to get enough nutrients in its body to produce all of those eggs. The males are free living. How they meet up with the females, nobody knows. But it's noticed us and it's popped back inside, and now it's just hanging out there, hoping not to make any currents or vibrations in the water. So that's also kind of a protective mechanism, not to make any movement so that a predator can chomp you up. We have some very good video of this from yesterday, so you can refer back to that. The alien from Aliens, the alien queen. It looks like this barrel is again a pyrostome barrel, which is quite interesting because we haven't seen any pyrosomes that were not barrels yet. Any free living pyrosomes. They must be here because the astronomers have found them. Let's see if we'll be able to find any with the SuBastian as well. Okay. Got a couple of good money shots there. So where are the pyrosomes these guys are making their barrels from? Fish, maybe a cyclophone. Another fish. Cyclophone. That was either a rhizarian or a Lavatian house. We'll have to look at the original video later. Oh, there's a hydromedusa. Looks like a trachymedusa. Possibly an arctopodema. In focus shot, see if we can see where the gonads are. Just eject its tentacles there. It's a repellonematid trachymedusa. If there's a muscle-free zone around the stomach, which is circular, then we probably have an arctopodema. I don't know if we can get a tentacle count on that guy, but it looks like it's got quite a few. Arctopodema normally has hundreds, so you won't actually be able to count them. Can't quite see with this yet. Looks like we've got a couple of in-focus shots from a few different angles, so hopefully that'll be enough to give it an ID later. I'll keep on heading down, see what else we can find. Try and characterize the water column to as deep as we can, while still keeping the quality we need to get species level IDs, because without knowing the species, and each species having different habitat preferences, the data is not as useful as it would otherwise be. Looks like we've got a fish, maybe even a myctophid. And a siphonophore, possibly a calicophoran. Another period. Okay. It also looked to have the most mature of the chlamydia fluorescing with green fluorescent protein, which I'm sure I've seen in a couple of the other individuals we've been videoing up until now in this dive. It's interesting why it wouldn't have the fluorescence in the rest of the chlamydia, and only the most mature ones, which are left over from the larval stage. Perhaps the green fluorescent protein serves another function in development. Coming down to 495 meters soon. Wow. Just passed through a swarm of plankton there.
Just a quick insert here showing where we are. We're about 4 kilometres away from the Osprey Reef. We can actually see the waves breaking on the edge of the reef. Very nice day here in Australia, very flat seas. But we're at that very big lump, the big boulders on that lower slope, right in the centre, lower centre of the inset. We'll have a closer look at that for the after Dhugal's finished with his midwater dive, and we'll take you on a ride over those blocks. Thanks, Viet.
What do we have here? Is this a Siphonophore?
Yep.
Yeah, I can see the flashing of the comb rows. Oh, it's just pulled its lobes in. So I can see 2 white spots, which are the tentacle bulbs probably. Are they placed at the top of the stomach rather than around the mouth? Is one of them a food item? If the tentacle bulbs are placed around the top of the stomach, the arboreal end, then it will be a Bathysceroe. If they're placed around the mouth, it could be a number of different things. If the gut is pigmented, it may be a Lampectase. Is that the same animal? Or a different one?
Same one.
Same one. So we have it in its relaxed state and its reactive state. They're quite small.
Yeah, also driving into the camera, so it's tricky for us to get it in focus.
Right. It looks Bathysceroe-like. I think those tentacles were positioned at the top of the stomach. It doesn't swim like Fosteri, so this may be the one, could be Paragaster that we've seen on a few of the other dives. Hopefully that video will be good enough to get an ID for it later. And we probably should keep heading down. I think we're running out of time.
Yeah, about 30 minutes left there, Dhugal.
Roger. Let's keep on heading down. It's a Sormissus, I think. Larvation. Nice. You can get a tentacle count from that. That's good. And now please turn bottom up.
I'm counting about 26 tentacles.
Wow. And stomach pouches at the base of each of those tentacles, I think. Be nice if you turn around a little bit for us. That's good. You get the stomach pouch, the shape of the stomach pouch. I can't see any gonads, which are normally on the rims of the stomach pouches, just near where the tentacles are inserted into the bell. So it might not yet be a fully mature animal. Okay, that's good. Okay, great.
There's a section of tentacles. There's a section of tentacles that are closer together than the others. What do you think causes that?
Yeah. I think something's probably chomped it and it's growing them back. Something probably chomped the bite out of the rim of the bell.
Right.
Normally, they, as they grow, we think the number of tentacles increases by in between each developed stomach pouches, as a smaller stomach pouch and a small tentacle form. So you have one short tentacle. And this may be where they get that posture of having one tentacle points up, the other one points down, and then comes down. It may be related to development.
Have you been able to get 20 of one species and put them in size order and see that the tentacle number increases with age or size?
Another Sormissus here. Yes, we use all the animals that we have samples of and graph tentacle size versus tentacle number versus bell diameter. And there's a disjunction, so we're pretty sure they're not developmental stages of the same species. They're not overlapping at the same. They have the same tentacle number at different bell diameters, for example. And we've done DNA work on them to show they are in different clades. So we just need to define the morphology other than for the one with the large number of tentacles and then the one with the smaller number. We can tell them apart, but it looks like there are species hidden within the smaller number of tentacle morphs as well. We don't know how to tease them apart at present. So hopefully these samples will help us to do that.
Right. When specimens have few features that overlap, that tends to be the challenge, isn't it? Is finding those informative features that separate species.
Yeah. It looks like it has placement number, form, could be important, but not all of the animals are mature. And depending on the state of maturity, the shape, etc., is different. So still looking for a better character for that.
Yeah. Same with the corals. You know, if they're young, it's hard to tell if it's small because it's a small species or small because it's young.
Yeah.
And the same with branching. You know, we think we know how many branches and how many spines are in the species, but it seems as the branch gets bigger, there's just more rows of spines. You know, we have these features, but they're probably wrong. Once we get enough of one species of different size, different ages, we'll probably be able to, you know, stop counting rows of spines and things like that.
Was that in Tasca?
Siphonophore coming into view.
Plasticity and morphology for cnidarians. It looked like a lobo. There he is.
Google, I was looking for a phylogenetic tree of all the jellies, and it seems most trees don't really have most of the species. How many species are there that you know of?
6,000?
And how many have been sequenced? How many species that you're sure are that species? Because
Oh, you like to ask difficult questions. Yes, a very small number compared to that.
yeah. And how many of the species that have been sequenced are probably incorrectly named as species and misleading?
That I know of right now, only like 4 or 5, but I haven't checked all of them. And some of those sequences are actually from contaminants like protists and ostracods rather than jellyfish even. But that's GenBank for you. Yeah, that's why we are very careful to only have gold standard sequences in our analyses where we have the specimen to refer back to. This is a scalia siphonophore. Morphologically, that looks like Asymmetrica. Could be a damaged Formosa, but I don't think the neck discern is tapering enough for that. Get a side on view. Yeah, that's Scalia asymmetrica. So our Scalia species are distributing with different species in different depth layers, which I guess is what you'd expect from ecological hypotheses. Mind you, we still only have like n equals 1 or 2 for each.
It's like only 1 or 2 specimens total, right?
Yes, but every data point adds an extra specimen to infer things like that. Thank you for that. 525 meters.
Yeah, 7 degrees Celsius now at 537 meters.
Roger. Oh, what do we got here? That overlooking thing in the foreground. That might be the nectophores. Yeah, it looks like it's shed its tail. It sensed us coming and got out of there. If you can zoom in on those nectophores and see if it has bulbous stomatocysts to see whether it's Desmophes, which is the animal we caught earlier. Kind of hard to tell with my video. I thought I just saw a projection into the mesoglia that was rounded, but maybe not.
Dougal, question: are they those nectophores? They're not coordinated in their swimming, are they? They're just independently pulsing. Sometimes they look in sync, and other times they look like they're just doing it whenever they randomly.
Yeah, good question, Rob. Thank you for that. They can. I don't know about this particular species, but in some they certainly seem to be in sync when they're swimming. But yeah, there's no one really who's ever put an electrode in there and done work. It's hard enough to get the animals up to the surface in one piece, and we're still sorting out the taxonomy in a lot of cases. So there's a lot of work to be done. And now in the age of grey logic and soft materials, if we want to be designing a new underwater robot, then we've got things like this to look at as possible ways to propel it through the water. So it's work that really should be done. It would be very interesting to see.
Yeah, I agree. There's all sorts of very clever biomimicry robotics, looking like fish or jellies pulsing. Yeah, it's quite interesting to see what's coming out of robotics labs.
Krill. Seems to me like there's fewer krill than there were a little bit shallower. Might have gone through the main layer. Still seem to be a few jellies around though. So down at 570 meters, it wouldn't surprise me to see some Colobanema kicking in somewhere soon. We've seen them on the last dive. Oh, what's that? That's a shrimp? Tomopterid? The antennae look to be coming out curved rather than sharply elbowed.
Looks copepod-like.
It's a copepod. That's a big copepod.
Oh, may even be a polychaete. I don't know. We'll get. We'll
That's a
zoom in.
tomopterid polychaete. A swimming tomopterid polychaete. Some of them carry their babies on their tails, that long stringy projection coming out the back of the animal.
Yeah, fast swimmer, this one.
We've seen this whole cruise. Cool. Yeah, I don't think you'll catch him. Once they know you're coming, they're like, woo! We should keep heading down to make sure we get as deep as we can at this nice slow speed.
Dougal got a question here. What happens when a siphonophore breaks apart? Do the pieces form their own siphonophores or die?
The budding zones, depending on the siphonophore group, the budding zones are between the float and the upper nectars is one budding zone, and this produces the zooids on the nectosome, so the swimming bells, and for aphelinids, for example, the nectosome tentacles. And then there's a second budding zone at the base, the lower end of the nectosome, and this is where all the familial units are budded off from. So all the things on the tail, like the stomachs and the bracts and the reproductive organs, etc., the palpon feelers, are all, wow! Is that a siphonophore or like a live one or a dead? Could that be a butterfly system? So yeah, as long as those budding areas are intact, they can reproduce more zooids from there. So if you're on the part of the siphonophore that doesn't contain the budding area, then you're on your own. You slowly sink down to the sea floor, eating and reproducing as you go, and like enjoying your final moments on Earth. And if you're on the front half of the siphonophore with budding zones, then you can keep reproducing asexually and adding more zooids to your swimming and floating colony in order to get back to full strength. Like that. Sounds like. In some siphonophores, the lower comidia will break off and live a free-swimming existence as something called a eudoxid, and that's the sexual stage of a calicophorine siphonophore. And they rotate between the polygastric, many-stomached asexual stage and the single-stomached eudoxid, which is the sexual stage. It's a very involved and complicated sex life. Some siphonophore colonies are both male and female at the same time, and some have separate sexes. Oh, that looks like a colobonema, that bluish tinge. It's probably a colobonema with its trailing very long tentacles behind it. Yep. So this is also a repellonematid trachymedusa, Colobonema sericium. See the very long, thin, white stomach, which hangs down about halfway down into the bell, and all the tentacles coming from the rim of the bell, and the velum, which is that membrane it uses for swimming, looking green right now, very large and developed. So this is a very fast swimmer. When you shock it, a predator attacks it, it normally will throw away a few tentacles, and these tentacles will glow in the dark as kind of a decoy that the predator will go after while the medusa itself swims away and tries to escape.
Dougal, is this rainbow jelly that you talked about the other day that's named in Japan? Because it had really beautiful iridescence on the bell.
That's it, yes. The musculature on the internal surface of the bell is very highly developed and refringent to light. So depending on the angle of the light, you get reds and greens, and really beautiful animals to photograph in a tank back on board ship if you have the right setup. the rainbow jellyfish in Japanese.
So I am paying attention.
Ah, yes. You'll be a trained midwater biologist soon. In fact, you probably know more about midwater biology, like surveys with ROVs, than there's only a handful of people in the world who do this kind of work. It's still very largely unexplored. Very few surveys to do it, and we know the most about the midwater region from the Monterey Bay Aquarium Research Institute's work in Monterey Bay off California. And then we have information off Japan from some of the work we've been doing at JAMSTEC. And the other information is very, very few and far between. There's a little bit from up off Woods Hole up in the northeastern US. And some recent work with the Oceanus Explorer, for example, in the Pacific, but really not large amounts of data when you consider how large the open ocean area is. We have selfs here, a self chain, it looks like. Good solmissus food. The white spots are the stomachs. So we have 1, 2, 3, 4, 5, 6 animals here. Is it 6? Joined together. The way in which the individual selves are joined to each other gives us a clue as to what the species are. If they're joined so they're lying next to each other and swimming through the water like this, it might be a pagaya, for example. If they're in a large colony, which is helicoidal, it's a helicosalpa. Circular, attached by filaments to a central spoke, it's cyclosalpa. Inland end, it's probably the genus Salpa, so this will give us an idea of which genus they belong to. Salps, sometimes there's transport to other animals, saccharine in copepods and known to attach to them and hitch a ride. And there's a possibility that lobster larvae or phylosomas also could attach to the surfaces of these cells and then use them, chomp on them while they're using them for transport as well.
5 more minutes, Dougal, and then we're going to have to kick it down to the seabed. We lost an hour this morning, unfortunately, so yeah.
You may just explain the crickets in the background. It's actually Japanese crickets where you are, not some weird echo.
Yes, that's a cicada. The mating call of the cicada. I'm here in the mountains of Nagano Prefecture in Japan, by a lake, doing a survey off the east coast of Australia. Amazing what technology can do now. So we're here at 615 meters depth. I'm here out in the mountains in Japan with not so great Wi-Fi, but I'm still able to be looking in pretty much real time at the animals going past here and pushing back the boundaries of science as we do a midwater survey here in a place that has only really had one dedicated midwater dive here before.
So Dougal, we're wrapping up shortly. What's your highlight of this midwater dive today?
Today's. I'm actually a bit stoked finding that Siphonophore gilmeri, the Siphonophore that had all those little red dots, is another Colobolema, it looks like. Because that's the first one we've seen on any of the dives on the current campaign, and it's actually only the second one I've ever seen alive in my entire working life. So that's very nice. I don't have a sequence from him either. And it's only been recently described, I think, in the last 10 or so years. So that was really cool. Also, the Siphonophores, getting information on those because you can't catch them in nets. So it's the only way we're going to be getting information on these animals. And we saw a couple of Bathysaroi, I think. and very nice dipid, which is an undescribed genus and species. So each one of these video records gives us an extra record to compare to the other animals we think are the same, and then we can say definitively, no, it doesn't have this structure because it's a mutation or something. Every animal we saw had this same structure, therefore we'd like to erect a new species. While the callings on the end of this columella tentacles are amazing, aren't they? It's almost like when they have a copepod or something attached and then the copepod escapes suddenly. The coiling in the tentacle may help the tentacle to not break as the prey is trying to escape, because Collemema ejects its own tentacles like a lizard throws away its tail. So they probably detach rather easily. So without that coiling, perhaps the prey can pull the tentacle hard enough that it comes off. So that might be an adaptation to that, like a trade-off between its defense and its predation, which is very cool. It's great to see these animals alive, where you can actually see that kind of behavior and hypothesize about why it might be so. So I guess it's time now that you guys need to head down to the seafloor. I'll still be watching, although I won't be yelling out, stop, please, anymore. But we'll still have the video recorders running and the cameras running. So if we see any large animals coming past, we'll still get distribution records for those. And very appreciated being able to have this interesting area of the world.
Thanks, Dhugal. Excellent, mate. We'll dial you in again in a few days as well. I'll send you the update of the program.
Roger that. So tomorrow's dive is a shallow benthic dive?
Correct.
Okay.
Here it is. Okay, I'll send you the update.
Thank you very much.
Great, excellent dive. Okay,
Thanks,
we'll head
Dhugal.
down to 1,500 metres.
Perfect.
Yes, we're heading directly down to the bottom now. Target depth is 1,500 metres. So the inset view just shows the 3D picture of Osprey Reef. Osprey Reef is that shallow area because it's artificially coloured. It's the reds and yellows. That's it's a large lagoon, about 30 kilometres long and at longest width and 8 kilometres wide. We're on a very large block in the centre lower portion of that inset, diving down on the seaward side. 1,500 metres, and we'll climb up a 200 metre high wall, 1,500 metres, and then go over the top. So spending the rest of the day in over a kilometre depth. Just for people watching ashore, we're just doing a live video broadcast with the Australian National Maritime Museum. So we have mobile video inside the control room. That's why we're quiet at the moment to let the ROV pilots talk to the video camera. J-Rod's just describing to the viewers the different sample boxes for this live feed to the Australian National Maritime Museum. Yeah, that's right, Brutal. It's a tech talk to the Australian National Maritime Museum, so we're going through all the different lighting, sampling, and all the archiving techniques on board the ROV. the numbered cylinders on the right-hand side of the ROV have our jellies. The ones on the left, they're our push cores. We actually push them into the sediment. Yeah, we thought it might be squidding, Dougal. We're aiming for 1,500 metres as a target depth, and then probably coming up to around 1,200 metres, so about another 150 metres to go.
Scientists on board.
See y'all later. Have a good day.
Bye-bye. Thanks. Excellent. And with us now, we have...
We're all clear, Tom's for 15 minutes. Sorry, guys.
No, that was great. I learned a bit there. Learned a lot. Good timing.
Come on the bottom.
No, we still need to keep going across.
We're coming to waypoint 1, Christopher.
Roger. Do you want me to come up higher into the blue water? Well, we did the waypoint 1 or stay?
Yeah, we're not quite there, are we? Yeah, let's do that. Let's move across. Thank you. Yeah.
All right, so
You get in here?
yeah, all right. So you know me to fly and look at stuff.
Nah, we'll just get over there.
Bridge knows we're moving.
Okay.
Chris,
Thank you.
No course down yet. No, we're just down here down the bottom.
I'll get down a little bit in case there's something crazy.
We've been traveling over pretty good. Just been keeping them up with the delta.
These were out this morning, but they came back.
Okay.
So, unfortunately, I won't have any excuse if I mess up on the flying car.
feet. Well, good afternoon, everybody. Scott Nichol back in the chair with Rob Beeman sitting next to me. We've clearly hit the seabed now on our dive adjacent to Osprey Reef out here in the Coral Sea Marine Park. So for the rest of the afternoon, we'll be taking you on a exploratory dive along a lower section of the Osprey Reef. We're on its western side, and you've got an image on the screen right now showing you the multi-beam sonar 3D perspective view of Osprey Reef, looking at that western face and off into the north in the distance. So our profile will take us, we're quite deep, we're 1,500 metres or nearly, and we'll rise up to 1,200. Our target site today is a very large block. We're not entirely sure of the composition of that block. That's one of the reasons we've chosen this location, but we suspect it to be limestone, but it'd be great to see some outcrop to verify that and perhaps even sample it. In addition, this rock, it's a massive feature. It's going to take, it's about 1 kilometre across. In fact, we may not get across its whole extent in our dive, and it rises 200 to 300 metres above the seabed around it. So a massive feature, and we're quite confident it's hard ground, so therefore we'll provide potential habitat for a whole range of sessile fauna. So we're hopeful of seeing a bit of diverse seabed, benthic sea life this afternoon on this transect, and as usual we'll be doing our 500 metre standardised sub-transect on the toe. So right now we're just positioning SuBastian onto the start of that 1 kilometre survey line we'll follow, and already you can see we're still a little bit high above the seabed, but just coming through the darkness there's clearly some rocky material, rock outcrop, but still dominantly soft sandy seabed. So probably at the start of the dive we'll set the SuBastian down and as per usual take a short sediment push call.
I can't move forward and I can't go down. It's in a position. I was kind of doing it. It's only 70 meters away from the pond. Pull that tether, I guess, and then we need to get it. It's easy. Get fast truck. It's impossible.
And we've just changed out our ROV team. We've now got Chris and Ben on the controls, with Viet backing up on all the audio and the YouTube, Facebook stream. And behind us we have Jeremy on the event logger and Marty McNeil doing the squiddle logging. So we're just repositioning the ROV directly behind Falkor, so it's this dance between the ship at the surface and the ROV 1.5 kilometres down, and we're just moving onto our transect, which because of the really excellent 3D mapping that we do, we can see exactly where we want to go. So we've got a lot of debris down here. Yeah, a fair bit of marine snow still at this depth. Siphonophores, just got a glimpse of a fish in the background, so we're still a little high off the seafloor, but we'll be settling down fairly shortly. Maybe.
Literally, this is me full down right here.
Yeah.
Trying to go to the sea as hard as I can.
Okay.
What's this?
Yes, we agree with you, Jason. Geology drives biology.
That's in fact, yes, that's our creed. He loves a good rock.
Ooh, what's that?
Yeah, might be a wood coral. I could try to sit down right here. We're pretty close to.
Yeah, let's try it. Yeah, let's try that.
See if we can figure out
if you can.
why it's not in line at all.
Okay.
That wood coral's a good place to settle there if you're going to settle anywhere.
Yeah, I agree. Yeah, well, just below us.
See it.
We still haven't collected a certain genus of deep sea wood coral. and
Okay, right.
No reason why. It's weird that it's just not everywhere.
Okay.
So
Yeah, okay, we'll get a good viz now as we settle down.
Yeah, we're very close to the start of our transect, and already it's looking quite rocky, interspersed with soft sediment.
I can sit down and try in front, but I can't sit down right there. There's too many rocks.
Too many rocks. Find a parking spot here. That's soft ground.
So quite a few boulders lying around there, Scott.
Yeah.
Because
Sure are. Yeah.
because one of the questions we have is this very large lump on the seafloor that we're going to run up over. Is it fully intact, or is it just a big jumble of boulders? Or that's one of the things we might be able to figure out from this dive.
There we go. Some,
Oh,
your favourites. Your favourites there.
Yeah,
Rob?
giant isopod burrow. I do like these. I like seeing the live isopods in them too. We have seen that occasionally. If anyone was following the Visioning the Coral Sea expedition back in May, we came across a whole wall of these,
Thank you. Push call.
like a big chalk wall, and it was just filled with burrows like these. And we did see the living giant isopod within one or two of them.
The wombat holes of the ocean.
Yeah. Yep.
That's better.
That's good.
So,
That's nice.
We're going to do push forward.
Push call. Thank you, Chris.
but the other thing about these burrows is they're colonised by lots of other animals, and we've seen a real range of other creatures inside, everything from octopus to lobsters. I think yesterday we saw some fairly large crayfish, quite large crayfish sitting in them, brittle stars. We've seen fish. So they're quite an interesting little microhabitat in themselves.
Out comes the
Are we going to use
sampling tray. Yeah, some rocks up in the sonar. Yeah.
for just more feeding too? Go for it. Roger.
So a lot of familiar names on the chat this afternoon. Welcome back, everybody. But in case there's someone new, first-time viewer for the operations in the deep ocean of the RV Falkor and its ROV SuBastian, what we're about to do is to collect a sediment core, a short push core, using the mechanical arms of the SuBastian carrying over that short PVC tube, which we reuse every time. And it's a relatively straightforward exercise of just using the mechanical strength of the arm to push into the soft sediment. The top of the tube has a one-way valve in it that allows water to flow through, and then as we pull up, we create a little suction effect to help retain the core inside the tube.
Never knew that the zip tie was actually functioning to to equalize the pressure.
I didn't know that either.
Down.
I didn't know that either until now.
If we
Yeah.
forget to do that, you'll see it because
It implodes.
It'll implode,
Yeah, it makes sense.
Clever. Did you go through different techniques until you were like, oh, just stick this?
Can you see the
Just keep it open. That's all you're doing. Yeah.
No, it was already come up with that one.
Oh, okay.
So that's a very successful core there. Full recovery, no loss. Always a good win to start a dive off with that. Good afternoon, Emily. Welcome back again. One of our local Australian viewers.
I'm doing good, don't worry. I'll try. I just did the push work.
Good. So we'll do another core standard practice here. We take a duplicate or replicate core just to give us more material to work with back in the labs. We can use these sediments to look at the sediment texture, get a better understanding of the energy that's been involved in transporting these sediment grains to this location, but also the composition to understand the source. Same spot, thanks Chris. Yeah, that's fine. Yep. It's not absolutely critical where we take the core from, because we can tell looking across the seabed here it's quite uniform, so we have confidence that the sample location we pick, it will be highly representative of this environment that we're sitting on right now down at 1490 metres.
Good question.
What's the question here? Do giant isopods protect their young? Good question. I'll defer to an isopod expert on that. Maybe someone in the chat knows.
I wonder how big a juvenile giant isopod is.
Yeah.
Are they still pretty giant?
Yeah. Thank you, gentlemen. That's a good start for the profile. Now there was a potential coral.
Oh, where was the whip? Is it behind us?
It's behind us. Yeah, I can spin around now.
That's right. We could head towards the. I'm sure it seems like this area probably will have a lot of sediment grabbing corals, filter feeders. So I assume on that rock it'll be filled with stuff. This is a strong current.
It was whenever I was flying over, but it's calmed down now. When I was first flying over, couldn't make any headway, couldn't get down for anything. But I think that had more to do with where our umbilical was.
Yeah. Dragon.
And our umbilical was getting pulled through, so then it sent down a lot of through. Alright.
Okay.
Camera view. Nice rock right here.
There is an ice rock right there. Zoom out. Okay. There's a couple
question
of
I think about the core.
cup coral and the. Well, we should. Since we're at the base, Rob, I think we should look at the base of our outcrop. If we're looking at. Let's just take a quick view at this outcrop.
Yeah,
It
looks
used to
like,
come in a manganese crust.
yeah, this dark staining is very common in the deep ocean. We see it starting kicking in around about that 400 metre mark. It's just a very light brown, and then it just gets darker and darker as you go down. When you get down to around 3,000 metres depth, it forms a very thick crust, and last year we were on expedition with doing rock dredges to around 4,000 metres, and when you pull up a rock, there'd be even up to like 10 centimetres thick of this black ferromanganese oxide covering.
Yeah.
So this is There's a hexagonal sponge. Doesn't look live to me, but it's quite.
Dead one higher up.
And yeah, big like a vase sponge. A lot of sediment on it. Looks like it's been knocked over.
I think we'll find a lot as we go up.
Yeah, so it's collapsed off.
Yeah. So it's almost a bit like a scree slope with a lot of sediment on top. I just saw a big fish shadow off into the right here. Yeah.
1,400.
Looks like a debris field to me,
It
Scott.
does. It's a scree slope for sure.
Lots of different
loose.
size.
So maybe we could.
Yeah, large boulders, small ones. That suggests it's not well sorted. It's just like a catastrophic collapse, and you just get a huge range of different clast or rock sizes in amongst it.
Different degrees of angularity and roundness. Yeah, it just tumbled down the slope, pretty clear. Could grab one. Just to.
Yeah, there are plenty. There's lots of smaller ones here.
Yeah, okay. Yeah, okay.
Just seem to be lying there dead.
Jump over there and have your pick.
Yeah.
So we're at the base of a fairly steep rise here, and just getting a sense of what the seafloor is like.
Might be able to come down a little bit at the very least. Be alright with that.
Rather than try and break anything off, we'll just take a loose sample, I think. Just to see what the lithology is and nothing more than that.
Yeah, but I mean, once we can actually put these, if this is hard rock, which it looks like, we can actually put it through a rock saw, cut it in half. Of course, we'll now have a little dark crust on the outside, but we can see what the internal lithology is like, what it's actually made of.
Something loose. You can see down below us, Chris, there's any one of those loose, if they are loose. They look. Yeah, pick a rock.
Yeah, good question about the crust. The crust only forms where the rock is actually exposed to water, and so we've seen places where sand has been washed away, and the rock that the sand was hard up against doesn't have the same amount of manganese crust on it. So it's quite clear that where it's dark and black, it's been exposed to the ocean water for a long time.
Yeah. Let's give it a nudge. Give it a tap.
Will this go in Biobox 1A?
Yeah, thank you. You get one? Oh, yeah.
There we go. Yeah.
That'll do. That will do.
Yeah, so even though this rock is not an in situ outcrop, which is always the ideal, just taking a piece now, we can see that it's part of a big scree slope, and that can, I guess, tell us something about, I guess, all the different processes that happen on this particular coral reef, like underwater landslides. So it looks like we're on a big scree slope, big boulder debris zone, and a piece of this rock should actually be, well, we believe it would be originally limestone, but we can actually cut it open and have a look.
that's great, actually. Just the right size.
Yeah, nice big solid piece of rock.
Not too heavy to take home. Good. That looks, it's got a cover of the fine sediment on it. That feels right.
A's or any others? Any of these? 1A, please. Only because I already
Written it down.
Yeah, I'm new with this. I think you're good.
So of course we've got plenty of geologists, biologists on board, and we've got Jeremy in the control room. He's very keen to get some more black coral, so we're on the lookout for deep water corals while we're down here.
Absolutely.
And we've got permits to collect corals while we're out here in the Coral Sea.
Yes, thank you to Parks Australia, who are the government authority within Australia that have the responsibility for managing the marine park network nationwide. So our friends and colleagues in Parks Australia have worked with us to help set up and design this voyage, and that extended to issuing the permit to work within this area that is a marine protected area. One of, is the largest marine protected area within the Australian marine estate, the Coral Sea Marine Park.
That's right. The Coral Sea Marine Park is nearly 1 million square kilometres, and we're at Osprey Reef in a very large feature of the Coral Sea Marine Park. It's called the Queensland Plateau. Now, the Queensland Plateau itself is around 260,000 square kilometres, so it's almost a quarter of the size of the Coral Sea Marine Park. And Osprey Reef, where we are now, is at the very northwestern end of that Queensland Plateau. Scott talked about the work we did with Parks Australia to help bring this expedition about, and it extended to try to find the best places to visit in the Coral Sea Marine Park, because there's just on the Queensland Plateau itself there's over 30 large atolls, coral reef atolls, shallow reefs, and Osprey Reef is often quoted as the jewel in the crown because of how great it is for scuba diving, and also for, I guess, as we're finding out, the deep marine life that's here.
Urchin?
Big urchin here, and something long up ahead. What's that?
Something long, lying flat on the seabed there, yeah. We should have a look at that just to establish. Is it? Yeah. May not be a. Maybe anthropogenic. Let's see.
Oh, it looks like a tree branch, really.
Or a bit of bamboo.
An odd place to find it. I mean, we're over 170 kilometres away from the mainland. It does look like a piece of bamboo.
Bamboo.
Yeah. Possible.
Let's just say that floated around and here.
Well, we have the South Equatorial Current that sweeps in out of the Pacific, so a lot of material, coconuts and vegetation do float in from east of us. We have Vanuatu and New Caledonia. These are two island nations to the east of us, though they're several thousand kilometres away.
Yeah.
We found a coconut, didn't we?
Yeah, a few days ago in Canyon.
Yeah.
That was neat.
So we know they float up onto beaches in North Queensland, but they're also found in the depths. They just get waterlogged and fall down. Oh, what have we got here?
Jeremy.
Is this a stalk crinoid or?
Yeah, let's look at this depth of this bad boy. Beautiful.
So this is a feather star. The question is it a comatulid, which means these short-legged ones, and they can just grab onto rocks or anything that's high in the water column, or is it a stalked crinoid, a sea lily? We'll be able to see pretty quickly as soon as we come around.
Yeah, you can come on top of it. You can look down.
Oh yeah, this is a sea lily, a stalked crinoid, and these are particularly interesting. These are relict fauna. They have they're in the fossil record going back hundreds of millions of years, and here in the deep coral sea, they are living on the sides of Osprey Reef. So I mentioned comatulid crinoids. They're cousins. They've got short legs and they can actually move around and even swim. But these have to attach. So these, they have beautiful colours and they sessile. They don't go anywhere, right? So this is a classic sea lily or stalked crinoid.
Beautiful.
Nice.
Yeah, beautiful. So there's west of Cairns where I live, there's a Devonian coral reef cave system called the Chiligo Caves, and you can actually walk over fossils 400 million years old made up of stalked crinoids. So the actual stalk of them, when they die, they break up and they make up a very big part of the fossil record. And just inland from Cairns where I live is you can walk over just kilometres of these stalked crinoid discs.
We still haven't found the fossil black coral.
Are they?
But they should exist.
Okay.
I don't think the people that are looking know what to look for black coral, so maybe one day you can take me there, we can go searching for the first fossil black coral.
Are they similar size, Rob, to today's crinoids?
Yeah. The stem can be a little bit thicker. Like these living relict ones that we're seeing here, they're quite thin stem, probably only about maybe 5 millimetres across. The stems of the sea lilies we see in that sort of Devonian reef tend to be a little bit wider. But when they die, they tend to break up, but they have this classic shape. They're very easily identified.
Yeah, okay.
Cool. And look at that. I mean, that is a really obvious big debris field, isn't it?
Yeah, this is absolutely what it is.
We got
Still,
Bubblegum?
we're still coming up.
or maybe even precious coral. Could it be corallium? So we do find corallium here. That's precious coral. It's a pink coral. Not sure if that's what we're looking at here, but we have found them in the deep waters of Osprey Reef previously.
Looks like an octo coral. Be a good one to get.
Okay.
Yeah. Can we turn the lights down a little bit? Just to get there.
I think you have lower lights off there.
Well done. That helps.
Are the lasers on?
Lasers on. Lasers on.
Yeah, if it was a corallium or precious coral, when they die, they still retain that beautiful pink coloration. Hard to tell what we're looking at here. It looks octocoral-like to me, doesn't it?
Yeah. I think, and we'll know once we put metallic fingers on it, how if it's soft or if it's pretty solid. But it looks like a gorgonian. Okay.
And just above it, what do we got here? Is it like a polychaete tube just above it? It's got its tentacles out, whatever it is.
Yeah, maybe. Okay, we can grab this. Sorry for reaching over.
No, that's good.
Okay, we're going to try and get a sample of this.
Different approaching.
No, I don't think so. It looks like an octocoral of some kind. Slimy schmaltz. Hydroids on the dead portion. Yeah, right. Yeah.
That's a good one.
Big rubbly slope. We're at the base
So
of
we're
this
still-
big cliff, right?
Yeah.
Are we just looking at the boulders at the base of this
I
big
think we
cliff?
are. We really haven't come to the cliff proper, I don't think.
We could put this
Another
in
100
one
metres at
rock.
least before we
It's a big rock, but still
might start to see a sheer wall.
Imagine how dramatic it would have been to see all of these big boulders tumbling down like this. Let's hope that's not going to happen here.
Yeah, well, that's right.
But I mean, look at the size of them. I mean, who knows how old this is? But it is a feature of like all the mapping that we've done around these 30 reefs have found really large undersea landslides.
You're right. I mean, they're not cemented together as clasts, so they're supporting each other, as it were.
To give you a little bit of
Framework support.
When we come down below and we reach up, that might be a better option because right now it's wide or this time squeeze. Like this, but alright, let's see. Bring your shoulder down, and then wow, bring the toe up underneath it.
putting them to the test here.
Got 70 years of molecular experience sitting next to me, so.
Did you say 70?
I did say 70.
Sounds about right.
This is kind of an awkward spot.
Looks hard.
Yeah, we're using their right arm coming in over the top and underneath.
Onto it right now.
Yeah, you got a bit of it.
It's just dropped
I was
out.
going to say just
blind
it's harder than I
sampling.
thought it would be.
I've never seen blind sampling.
Still get that one.
Going by feel, but you know, you're down at nearly 1,500 metres. It's tricky.
Interesting.
Bring your shoulder down and elbow up. Put one underneath it. See it better. Underneath. I think this one you can grab tight.
It's a really
How long that
hard.
small piece go, right?
You can, if you can get the piece on the ground, that'd be fine. Or if you want to get what's still,
Hey, the one on the ground's easy, probably.
there you go. And we keep the coral alive.
Alright, hold on a second.
Win-win.
Oh, that one? Is that piece right there? Oh, it just dropped. That piece right there, right?
And although we didn't mean to collect only a part of the colony, since we're leaving the part that's still attached to the rock alive, it'll continue to grow.
Bring your shoulder down,
I wonder what that is.
down, elbow up, elbow out.
Looks like bubble gum, you know.
But yeah, but it's hard. You know,
It is hard.
like it looked. I mean, it broke like a stick. Like,
Yeah. So precious coral tends to be a bit redder in colour.
yeah, right, like
Yeah.
like rumblum. Yeah, but there's pink coral as well.
Right, you got it. Got it. I'm gonna close slightly.
We'll get an answer soon. Kathy will be able to help us out with that.
Well done. That's
He is the
With the fingertips.
that is amazing work between in that crevice
artist there.
with all those rocks around us.
It's actually a smaller piece over here too.
That's okay. One, just enough at this stage to have a physical sample will be enough. You can put it 1A.
With the rock.
Yeah.
Alright, I'm going to.
That's good.
Just open up, just open up. Shake it a little bit. Alright.
Maayo.
Okay,
Alright.
back up. Continue up our line. 3.2 and onwards.
So what's interesting, Scott, you notice the these hexactinellid corals,
Yep.
sponges,
Yep.
you know, they seem to be lying around, lots of them. So you think sand
Yeah,
has
debris.
come down and just knocked them over or buried
They
them?
come
and,
down with a little avalanche flow themselves.
Yeah.
It's a bit of both.
there's a fair bit of sand here.
Yeah.
You know, if you're a filter feeding animal like a sponge, you really need to be kept clear, don't you? But yeah, you see that there's quite a few just in amongst the rocks there. They don't look very healthy to me. They look,
Not at
they look
all.
dead.
And I guess the one that we collected was underneath the rock, which would present itself from.
Yeah.
Yep.
You can see the staining changes, perhaps where the sand has been washed away and it's not as well stained. You've got that browner colour where everything else is quite dark from the ferromanganese oxide staining. Wow,
That's a great landscape.
that is incredible.
This looks like the same coral we just collected. Maybe.
Very angular blocks.
Wow,
These are just
maybe we should have looked up before we pulled that rock out. Not that one. Not that one. Oh my God! Look at that. That is just hanging there,
precarious.
and we. Oh my God! Okay, are we on this really steep wall that we think we've mapped?
Yeah, I think we're getting to it.
Yeah.
Yeah.
Wow. Everything's just huge, all leaning on each other, holding each other up. And you're right, it doesn't look cemented, does it? It looks like it's just hanging there.
Yeah, this is precarious. I think we're safe, but but it's geologically precarious.
Wow. Just a massive landslide. Okay, we're hitting some sort of outcrop. Looks a little bit more solid here on the left. All right, could we assume that maybe the boulder debris has fallen off these walls?
That's certainly a source. That's it. Is the source. I'm thinking.
Yeah. It all seems to be leaning out, doesn't it? Or is that just the orientation of the ROV?
There's a Holothurian.
Can we zoom out to see the big picture?
Yeah.
Kind of see.
I guess pull back a bit. Do the trick. There's the camera.
Oh, there's a glass
sponge.
Yeah, that's a healthy one. I mean, we do get, this is cyclone country, but we're quite deep here, and it's very unlikely that cyclones would have, the wave base wouldn't come down and affect this kind of depth. But we have had cyclones across Osprey Reef and in the shallow coral itself, right in the waters that you could swim on. There have been quite large pieces of limestone coral knocked out of that, but not at this depth. This is something completely different.
Looks like it's just, if you look to the left, it just stops right there.
Right, okay. Yeah, so we're off the edge.
Interesting.
Spinning around to the left, everybody. So that's giving you a bit of a view along the outcrop.
Right.
And then turning back onto our line now. So that makes sense. So we'll continue to ascend. this slope. Great, we've got the inset map on the screen right now to give us that bigger picture perspective. In fact, what you're looking at there, that grayscale effect is what we call the backscatter data. That's a measure of the relative hardness of the seabed. So where it's brighter and lighter and whiter, that's harder. That's where our sonar signal is reflecting more strongly off the seabed. So that's as you read that, you can see it's a real
soft world,
a real patchwork pattern of hard and light, hard and soft, dark and light.
We collected, I think, 2 of them now. We're probably fine. They're working on the genus of this right now. There's a paper review describing it, or amending it.
So, go ahead, Rob.
Yeah, just the insert, just doing a slow spin around that, the red-orange area, that's the shallow coral reef. You can scuba dive around the edges of that. The length of the reef is about 26 kilometres long and about 8 kilometres wide, just to give you a sense of scale.
I'm curious if it's hard like the others. Maybe quick zoom in.
Yeah, go for it. There's quite a few on this little face.
Side view or
Yeah, as we glide past, we can see it the other way. If that isn't too disorienting.
not a problem.
So there'll be more we can look at.
Another one to the left as well. Everyone. With a big one, okay, then. And also the big.
And also the different. You mean the one in front of us now? Yeah, okay.
He's like that immediate gratification kind of guy.
Yeah, I'm with you.
And we've got Julia Summerling watching. Welcome aboard, Julia. Julia's probably done more scuba diving at Osprey Reef than anyone on the planet, so she has long experience working with dive tourism vessels out here, because Osprey Reef is one of the prime coral sea reefs for scuba diving.
We're just looking at it then, or everything needs to be snatched.
Yep, then.
So Scott was just talking about the backscatter. This gives us sort of a relative texture of the seafloor, and we can generally tell soft sediments like mud versus hard rock features, and we deliberately chose this place here to go over because one, it was a very large feature, big steep edged feature, and it also had a very high backscatter reflectance, which tells us that it was quite hard. So one of the great things about this expedition is we can both get that 3D picture, we can get the texture, but we have the camera to go down there and actually ground truth it to find out what it really is.
Just take a quick zoom of her.
Yeah.
Stop. That's okay. A little bit of sponge. And just even out. Square up. Sponge.
Yeah.
What's that?
It's an enemy.
I thought it was something exciting.
Yeah.
You must be thankful for this now.
Bit of discussion going on the YouTube chat about the influence of cyclones on this reef and whether the cyclones may have caused this landslide, and questions about how old is this landslide. So very difficult questions to answer, but certainly these waters are too deep. We're down at 1,460 metres, far too deep for a cyclone influence.
Just to let you know, we're making small balloons because we're climbing up.
Cyclones would be limited in their impact in terms of the physical disturbance, probably to the top 50 to 60 metres up on the upper end of the Osprey Reef water depths. Down here, it's gravity and time, which will lead to these landslide events.
Like earthquakes, perhaps.
And perhaps triggered by an individual seismic shock, which we do have. And establishing the age for this, very difficult. There's no direct way of
It
aging.
looks like a crab with an enemy on its back. Yeah.
You can age on land. There's a technique called cosmogenic dating, where you can get an age of the time at which a rock surface has been exposed to
it's got an enemy on its
energy,
back.
but I'm not aware of it being used in these settings. Amazing spotting, Rob. Yeah. Eagle eye.
I mean, when you look at the age of these reefs, these are quite old. We know that the whole Queensland Plateau was in these tropical waters as far back as 22 million years ago, and then there's been 3 large subsidence events at around 13.7 and about 2 million years ago. And so we do know that these reefs are really ancient. Now, over that long geological time, there's plenty of scope for earthquakes to affect the landscape. And if it's too steep, then you can just loosen all this rock and it just tumbles down. But that's something that we've seen around every one of these 30 coral reefs on the Queensland Plateau. There's a lot of boulder debris around the edges.
Another anemone?
Yeah. This is crab with an anemone on its back.
Supposed to be a costume party.
Yeah.
We'll get that little crabby hat thing. Because it's cute.
Okay. Let me know when you're stable, steady.
Right.
One fashionable crab.
Yeah.
Crab and drag.
It's a free ride for the anemone.
Yeah, I mean, a good question. Is it camouflage? I would think perhaps as defence. You got a stinging anemone on your back, nothing's going to come and grab you too quick, and they're going to get a mouthful of stinging cells. So that might be a reason why you would do it. Decorated crabs can do this sometimes just for camouflage. They just look like the background seaweeds and things like this. So I guess there's lots of reasons why they would actually put an anemone on their back. Oh yeah, this is nice. And it's got a brittle star wrapped in around it. Is this similar to the one we just took there, Jeremy?
This looks like it has zoanthids on it, doesn't it? Or those polyps that are just closed up. No longer confident, but James is online. He could help us out with that.
Yeah. What do you think, James?
It's like the thing getting better at it. Because you don't see any polyps open.
No, like little buds all over it. So what we're discussing is this could be either a living coral itself, a colony, or it could be the remains of, let's say, some kind of stalk colony that's died and has been taken over by another animal like a zoanthid. So we're just going to try and zoom in here, have a closer look.
Sit down.
I'm going to zoom out for a second, just to give you some. What does James say?
Zoantheria.
We're getting better at it.
Yeah.
James must be proud.
Watch this kids grow up.
That's right.
And look at it.
That's okay.
The other,
All right, we'll continue on.
Continue up to only getting to waypoint 2. Good.
Yeah. Okay. Starting to see the difference.
Yeah. Okay.
But it's amazing. It looks like it's a part of the coral. You know, it's a consistent red color, but it's the zoanthid tissue that's red, and they just fuse together to make one large colony.
Yeah, it's very confusing, isn't it? Yeah.
I mean, essentially that's what a coral is, you know, but these are individual zoanthids that have taken over the coral. Oh, what's this?
Ooh.
Yeah, okay. Something off to the
Okay.
left.
Something in the sit cam. The sit cam's a lot wider.
We'll get it for everyone in a
screen.
second
That's a
now.
cool looking bamboo.
Yeah, isn't it?
This is different than what we've seen so far. We're probably gonna have to take a branch of this.
Okay.
I don't have any more big plastic bags, so we have to limit. But
This is a really big bamboo coral.
yeah, let's try to get some good images.
Yeah. I tell you what, Jeremy, why don't you get in the hot seat?
Get a couple of branches on Tuesday.
Yeah, look at the nodes on this. They'd be at least 10 centimetres long. We're looking at it from the side perspective, but I think it looks quite wide when we come in on another angle. You can see all the living polyps going up the side. Yeah, I mean, look at these, look at the segments. That is, wow, that's beauty. It's got some squat lobsters on it as well.
Yeah, we've stumped James on this one, so that's good.
That's right, they are really old, these bamboo corals, because they live only in these deep waters. It's 3.1 degrees C, it's very, very cold. We're in Antarctic intermediate water here for sure. When they die, the stalks break up. And you have these the segments break up themselves. I remember the first time I actually pulled these up in a rock dredge. They're only fossil material. They look like dinosaur skeleton bones. You know, like you're pulling up individual vertebrae. That's perfect.
And you could see, and I don't know if I'm right when I say this every time, but the polyps look very regular in the way they're oriented. So I could see, you know, probably 4 rows of polyps. You know, one on each side, one in the back, one in the front.
So they're not helical, are they, James?
They're not spiral, no.
Jeremy, there's sort of one on opposite each other, and then about 90 degrees.
So there's 2 opposite rows, two pairs of opposite polyps.
Right.
2 pairs of opposite polyps.
Yeah. Yeah, and some of the branches are bare at the top.
So you could see the repeated gargonin and calcium carbonate nodes.
Wow, that is a beauty. Big squat lobster defending.
So it looks like one of those harp. I mean, I think there's a genus that's
Harp-like, harp-like shape. Yeah, it does look like a harp. It would be as big as that, wouldn't it? I mean, this is a couple of meters tall.
Yeah, so we could safely assume this is a pretty old coral.
Yeah, for sure.
You know, because they're slow growing.
Yeah. When you the segments are interesting. The fossil ones, if you cut them in half, they look like tree rings, right? It's concentric circles, and some researchers go to a lot of trouble to actually do a whole lot of isotope analysis. And of course, you go to the center of the segment, and that's the older part, and the modern part is on the outside. So you can actually, by using microprobes, you can actually get understand the age and how the water column, how the water has changed through the life of the animal.
Right.
Just by looking at the individual rings, just like you would tree ring analysis.
So Rob, we are going to collect some of this for Uscape. Tokyo to do exactly that.
Ah, okay. All right. So one of our collaborators is Yusuke Yokoyama from Japan, and a long-time collaborator, very interested in Great Barrier Reef and understanding, I guess, the water column, and we can... That water column chemistry is locked up in the skeleton of these deep water corals. So we've been taking samples for Yusuke during this expedition.
Yeah, we don't need too much. Yeah, I think maybe like two of those large ones.
From right down the base.
Yeah.
He's fantastic.
So I just want to take a quick, while you're settling, are you going to settle
I
that?
gotta figure out the best way to get
Okay.
get over to what the.
Is it right to keep zooming in?
Yeah.
All right. We're just going to look at some of the lower polyps.
I might be flying around and making it difficult for you.
Yeah,
So.
I'm up for the challenge. All right, you're making it challenging.
Sorry. Alright, let's see if I can come to the bottom left if I pull this thing down over here.
Okay, and just for your information, we'll probably grab here to get like two large branches. You can do the other side. You know what?
So we're going to try and get a bit of a sample of this one, are we? Okay.
Yeah.
I try to right here, have you just come up on it then?
So it's such a steep slope. It's not easy to put the ROV down.
Hold on. No.
There we go.
Alright.
That's a nice view.
Nice view, yeah.
So you can see
So
the,
do you want to just describe the skin on that there, or the outer flesh, Jeremy?
Yeah, well, I'm not an expert at all with this, but it's probably a thin layer of tissue that runs along the entire colony. And it's probably very thin. You can
So
see
easy
the
to rub
nodes.
off.
Yeah, so you see the nodes protruding beneath it.
And when they die, the segment is mostly white, but the node pieces have a real dark colour. So it's it's quite obvious when it's living and when it's dead.
Right. Can we lower the light just a little bit? Just to look at these bottom polyps. Yeah, that's perfect. That's a nice view. Okay. So let's grab the two most outer branches.
Wow, so big.
Alright, might do this. Either sit perhaps.
I can't look higher, but you got it.
Yeah, let me try to speak just a little bit here.
It's beautiful. It's big.
Alright, how's that? Huh? That's good. Okay. Keep sipping that. I don't know, thanks for let's go for it. No free flight. Well, I'm just
It'll be easy to
pushing it. Okay. Jeremy, can you look up? No, we can't.
I can't.
Look up. But maybe this. Okay. Give this up a little bit. Yeah. Alright. You want it below where he is for the 4 branches there? Or
Is that the
Maybe below.
just below, so we get those 2 branches.
you want the last 2 branches that are branching right above?
Yeah.
Yeah.
Alright. So go between branches 2 and 3.
That won't work. So yeah, you have to just use that bend for it again.
It's interesting how the middle stem seems to have broken and is regrowing.
Or just naturally splits three ways.
Or maybe it naturally. Yeah, that's interesting. Yeah, that's fine there. Yeah, that's interesting. So with age, that middle branch will become two branches as it widens.
That came off pretty quick.
Yeah,
Yeah, it's good. Wow,
it's like a magic trick. I guess you could put it in 3/4.
It's gonna break.
Oh, it's okay. We don't need it. We aren't interested in the branching pattern of these. We just kind of want the tissue samples at this stage. We see what it looks like. We're not collecting the whole colony, so it doesn't matter if we get a small piece or. Very nice. And then I saw in the distance. You're way ahead of me.
Alright, so that, as far as I can go, so I'm just going to try to see down somewhere.
Very nice.
Ooh, what do we got over here? Is this another,
This looks like a black coral. It's hard to tell
or another
from
bamboo coral? Just one of these single.
Yeah, my gut's saying black, but let's see. We'll take a close look at the polyps. Just the way that it's contorted.
Yeah, the bamboos tend to be a bit more fluffy-like from a distance, aren't they?
Yeah, you can see it. So we could do the nice comparison of the bamboo polyps. You see how they have like kind of a neck and then the head? Mm-hmm. But with the black corals, all you see is the polyp mouth right on the,
Right.
on the
Yeah, okay.
colony. So they don't have that protrusion.
Yeah.
Oh.
And it's a stalk crinoid in the background.
Yeah. Oh, you might be right. Looks, you could see maybe the bare skeleton being white at the base of this. Okay, we're just going to place the bamboo in the bio box, and then we'll check out what this coral is ahead of us. The jury's out. It's okay, as long as you get the base in, and then close it, whatever breaks off the other end is alright.
So how tall do you reckon that colony was? And this is about 1 metre across here, and that was just one branch of it.
So maybe over 2 meters?
Yeah, probably 2 metres, yeah.
But it's possible to see 5 meter tall black corals, bamboo corals for that matter, down in the deep. Essentially, they can grow as long as they're getting nutrients and enough energy to continue to grow. They'll keep growing. Someone's asking for a 10-meter coral.
10-meter coral?
Yeah. No, all right. Sorry. I would say no.
Well, it's one of the tallest I've seen out here.
10 meter.
No, this one.
Oh.
It's just...
Well, the whip corals can very easily, like the one we're about to see right now is probably 3 meters tall. Let's take a look at that.
I had to put things in that were soft. 5-6 meters long. Oh, much better. Much fun. That's the left your shoulder. That's it. That's fine. And it's taken like 45 minutes.
Get it to the foil.
Yeah.
Get slanted on.
I was thinking we'll go to 6. On deck is 6:30. Okay, so get it to a 5.
Okay.
So we need to push up.
Yeah. Okay.
Get to our 5.
We'll take just a quick look at this coral, and then you do the 500 meter. So if this is a black coral with polyps that go around the axis, it would be it's a target that we haven't yet collected. If it has polyps along one row, then we've collected a few of them so far. And if it's a bamboo coral, we've collected a few of those. Which Houdini coral? I think it's up.
I don't know, second.
Shrimp? A shrimp.
There it is.
Right inside.
Yeah, I think it's a bamboo coral now.
That's the one we just did, so it's to the right.
Up to the right.
Whoa, look at that. That is really tall.
So it's, yeah, it looks like a bamboo coral. Now you can see the fluffiness you were talking about earlier. Once we got a little bit closer. So you could see the skeleton's white. I don't see nodes, but, you can safely assume it's a bamboo. So we could just move on past this one, and we can start our transect. That's pretty long. I'm curious how big it is.
Yeah.
Can't zoom out anymore,
It goes
actually.
up and up, and there it is there. Yeah.
Let's call it 10 meters. You asked for it, we give it to you. Where's this guy?
Oh, you can. I'll sit back. I'll finish my tea.
All right.
Unless you want to go somewhere.
This is where the excitement is.
All right. Stop it. No. Oh, no, it's not yet. That's a good one.
Sure.
I mean, once you fly along, should just about up.
Oh.
Kind of put you in a hard position.
I'll just put it away.
Which would you like?
That's okay. We can get to the transect.
Great, you just updated your position.
It's a while.
Okay. So you can see what,
Another one of those anemone crabs.
Yeah.
the fashion. Rich constraint.
So we're really only at the very beginning of our transect today. It's about 1 kilometre long, and we started at 1,500 metres, and we're driving up over the up to around about 1,200 metres, and then back down the other side.
All right. Now we're talking. Okay. So what we've got here is an Alternatopathes. It's a genus, in the Schizopathidae. And we've been confused, the people working on the taxonomy of black corals for the last 50 years on this specific group, the Schizopathids, have been uncertain about, what physical characteristics separate the different species and even the different genera. And so this Alternatopathes genus is fairly new. It's just a few years old. And our question now is, how different is Alternatopathes from Bathypathes? And the main difference between the two is whether the pinnules, the repeated branches, are subopposite, meaning one on the left side it's in one place, and then on the right side it's just offset from the one below it, or if they're alternating. And the pinnules on this colony are alternating. So we have lots of specimens from both genera, but there's also different species in each of these genera. So our questions are, where are the taxonomic boundaries between species and between the genera? And now that we have a molecular tool that is proven to be kind of the magic bullet to try to really answer all the questions we're asking, all we need now are the specimens. And here we do have a perfect specimen. So the pinnules are alternating. The pinnules are coming out to the same distance at the bottom to the top, which would be different than if they were triangularly shaped, where the bottom pinnules are really long and then they get really short as they go up to the top. So this is certainly probably an undescribed species, but we have other specimens that look similar to this, so we'll add this to the collection of specimens that will be used to describe the species and define the taxonomic boundaries. So, exciting.
You could aim for a whole colony, Jeremy?
Yeah, we can put it, it's not too big, is it?
Not
So
10 meters, but it'll work.
I'll just take a quick view before I'll be quick, I promise. We'll wait for it to turn. Okay, it's hard to see from here, but they are alternating.
I have to focus manually.
All right, manual.
It's not right now. Exactly.
There we go. So, yep, they look
Alternating
to me. Alternatopathies. Yep, let's grab it. Nice. Try to take a quick view before you just snatch it. So this is the back of the colony. You can see the stem, but not the mouths, the feeding mouths or the polyps. And you can see it has tentacles along the stem. Okay. Yeah, go for it. And so the reason why we're going to collect the full colony here is because, well, one, it represents probably what is now not known to science, but two, the morphological features that we use to describe new species varies from the top to the bottom of the colony. So if we were to collect just one branch, we would be getting only a piece of the puzzle. So the branches, the stem, the base of the stem, these are all important features that we need to look closely at to identify it. So when he grabs it at the base, it should pluck off with the basal attachment. Okay. Then I'll get out of the chair so we can get to our transects. There you go.
Hey,
Nice.
got the whole thing.
Alright, where do you want to put it?
3/4 is fine.
3 and 4?
Jeremy, can you just repeat the name of that one?
Alternatopathies.
Alternata.
So alternate. There we go. Awesome. It's a good one. So yeah, this is a black coral.
Alright.
And you do need that basal plate, don't you?
Yeah.
That's all part of the understanding the entire morphology of the colony.
Right.
So it's not enough just to have a branch.
No, a branch wouldn't tell us what the skeletal spines, which are these small microscopic spines, less than a meter tall, that run along the whole colony. The spines on the branches are different than the spines on the stem, and they also change from the top to the bottom of the colony. And lastly, on the very base of the colony, right above the basal attachment, there's what we call striatum, which are these grooves or nodules that are impossible to see without a scanning electron microscope, but some species have them, some species don't, and they're probably characteristic of different species. So we have the perfect specimen to better understand the species boundaries in this odd, misunderstood, or what not so well known genus.
Oh, yeah, Chris Benton.
Yeah, okay. Sure. Right. Yeah, we only collect what we need to answer the questions that we're asking, and these questions are fundamental to conserving biodiversity. If we are unsure about what species are living in the Coral Sea and the Great Barrier Reef, how do we go about making informed conservation decisions? And just by looking at corals, you're unable to identify the species and sometimes even the genus. So the limited amount of sampling that we do provides decision makers with all the information they need to make informed conservation decisions that make a larger conservation impact or difference compared to where there is no intervention or marine reserve or regulations and rules.
Yeah, I reckon. Okay. About 5, yeah.
Yeah, so we've really only just got down on the seafloor only a little while ago. We haven't actually been very far, but we've got about 2 more hours in front of us because most of the earlier part of the dive was a Dhugal dive looking through the Midwater zone. But now we're going to spend the remaining 2 hours on the seafloor slowly climbing up this very, very steep rock feature. Well, we think it's this very steep feature. We're finding out that it is rock. That was our hypothesis. Most of what we've floated over now has just been a huge debris field. Although there is some fairly exposed cliffs coming up. We've already been up one. We may have another one directly in front of us now.
Glass sponge. This long stalk.
Yeah. Yes, this is a hexactinellid sponge, a siliceous sponge, and we see all different shapes, but quite often they're on these big stalks. When they die, those stalks are just big siliceous spicules, and they're quite amazing, really. The spicules are long, continuous, like great long spines twisted around each other. And the first time you and I actually ever saw this, Jeremy, we dredged up a deep one from around 3,000 metres deep, and I thought we'd actually cut through some internet cable because it just looked just like fibre optic cables.
Yeah, perfect. Intertwined like it was done with a machine.
Yeah, it was like it was about as thick as your finger, but it was nearly a metre long.
At least flew over a live version of that sponge that we were seeing down below. That version looks like vertebrate. It's live and it's just there. So.
Like the base sponge?
I don't know. It looks like a vertebrate to me.
Some more of this pink coral that we've looks like we sampled earlier.
Right.
Ooh, what do we got over here? This is Anthomastus.
Oh,
This is a soft coral.
Yeah, right here. We've collected a few of these now.
Yeah, for your colleague Kathy McFadden.
Yep.
Yeah, Gorgon's head coral. They're octocoral, but they have this really. Oh, there's looks like a big bamboo coral in front of us.
All right. How many? Let's just try to approximate how tall it is. Have the lasers on? Yeah. Oh, wow. It's big.
Yeah. Okay. So you can see the lasers in the center of the screen. They're 10 centimeters long. Oh yeah, that's really tall.
I mean, it's huge.
2-3 meters.
I can't zoom out enough to.
Do.
Yeah, I'm saying it's probably close to 3 meters. Between 2 and 3 meters.
Could be bigger.
Ooh, is a jelly coming in?
Medusa.
Yep. Deep water jelly. They've got these really dark scarlet kind of.
What?
So he's going over with the ROV and measuring the altitude. Very smart.
Oh yeah, right.
So over 3 meters.
Yeah, 3.5 meters.
Yeah, there you go. We just use the altimeter, which is a little sonar, like a little depth sounder, on the ROV. Went over the top, so about 3 meters.
Okay. And the joints and the underlying.
Still very rocky, very rubbly. It looks to me like a big debris field still. I haven't seen a huge amount of just exposed outcrop. You know, that might be the origin of this boulder field. But we're still going up this transect that's going to run over the top. So it's like a big hill on the seafloor, and we're very curious to what it was made of. And we can see there's a lot of boulder debris, all different sizes, tumbled down on each other, interspersed with soft sediment. And this soft sediment would be calcareous sand from the shallower coral reef that's about 3 to 4 kilometres away from where we are now. So the stalk crinoid on the right.
Sometimes the shadows can be misleading. You know, it looks like a branched colony. When it's just There's another small one.
More stalk crinoids coming up. Sea lilies. Yeah.
Just updated your position.
The arms are long relative to the size of its body.
Yeah, like there were two different kinds. There was that very red one. I think we've got a species identified in the chat, and then there's sort of a yellowy, like this one, yeah, more of a yellow colour. Don't know if it's the same species, just different coloration, or no, it is different, isn't it? They're more. The arms seem a bit more sparse, a bit more spindly.
Yeah, this one just has 4 arms, doesn't it? But do they all have 4?
Well, they have pentaradial symmetry, but they can lose, they can lose an arm for whatever reason. Animals attack it or it gets damaged.
There's a lot out here. Oh, look, there's 2.
Oh yeah, one of these nice red ones. Oh, you're down in the deep sea when you see these ones.
There's another.
Yeah, on the right. Yeah, this is great.
Back there.
Yeah, I really like looking at them. It sort of reminds me like what the ocean would have looked like, 400 million years ago, and these things would have just carpeted the seafloor. It's pretty dense now here, though.
And you can see they have different lengths. Some of them potentially they're growing new arms as they get older and larger.
Yeah.
Like
and so they're like a series of discs all stacked up on each other. If we were to look in close, there's a dead bamboo coral over here, but if you look at the stalk, you can see they're thin discs all stacked on each other. And when they die, those discs they all break up, disarticulate. That's typically what you find in the fossil record. I haven't seen too many actually all joined together still.
Oh, it's got really long, small arms compared to its extent.
Yeah. Oh, there's more coming up. Yeah, actually pretty common here. Look, over here on the right-hand side, there's more.
Yeah. So we're seeing 2 different colors, and some with short arms, some with long arms. maybe there's some correlation with number of arms and age.
Yeah.
We don't know much about these stock runners, I bet someone does.
Yeah. Yeah. But quite amazing. Just attached to the side of the rocks on some small boulders or even bigger ones. Oh, look in the background here. There's more.
Is this our own, what do they call it? Land of the weird? Or?
Yeah. Yeah.
Exactly. There it is. Yeah, it does.
So we've done 2 dives here previously during the visioning of the Coral Sea voyage with the Falkor back in May and June, and I remember that like the first 2 dives was actually at here on Osprey Reef, and yeah, we got to see some of these sea lilies, but actually quite common here in this spot. Lots of interesting imprints on the seafloor, and we see these quite regularly, but I do not know what they are. They're kind of clusters of regular patterns of burrows. Right? They're not like imprints left on the seafloor by, let's say, an echinoderm like a sea star or something. These actually have holes inside them, so we're not really sure what they are, but they're very, very common.
They're going to pull down a little bit. It's 175 meters away from here now. And speed them up. So I go back and get these.
Okay, is this better?
No, we can't hear you hardly at all there.
We can hardly hear you.
So there's a comment about maybe giant forams making these holes.
Do not know.
There you go.
Thing is, they're burrows, and they have this distinct pattern. You know, it's like parallel lines of holes, kind of circular, but you can see that they definitely have a parallel shape. You know, it could be a worm coming up with different burrows, but you know, the same worm is using different holes to connect with the water and draw in water and food. Don't know.
We got pretty far away from the ship.
Oh yes, crustacean could make them. So if you're diving, scuba diving in the shallow lagoon of the coral reef, say Osprey Reef, which is only 3-4 kilometres away, you can see burrows on the sandy lagoon floor, often made by crustaceans. But you know, we just haven't seen. I guess we haven't seen the animal that makes them, so it's still a bit of a guess.
Back about 120. Carry on.
It's good. Yeah.
So we need to wait for Falkor to catch up to us.
Yeah, this is 175 years away.
We're just repositioning the ship to where the ROV is before we start our transect.
So we're ready. Still
Do you remember what you saw on the last trip when you did the dive a little bit shallower than this?
Yeah, well, we saw sea lilies, not as many as we've seen now. It's very dark rock, this ferromanganese covered rock. Lots of hexactinellid corals, these siliceous, siliceous sponges. And of course, lots of nautilus.
Okay.
This is nautilus country, and we stop for nautilus.
Yes, we do.
Yeah.
But is this too deep for them here?
I think so, yeah. So we got to see a lot of nautilus in the last expedition. Typically, they were in this zone between around
sort of 400 to 800 metres during the daytime. During night, we know that they vertically migrate higher in the water column, but here we are down at 1400 metres, and I think this is just too deep for Nautilus, although, happy to be proven wrong. But yeah, that's just not the sort of, this just seems a bit too deep for just what we've seen so far. I mean, it's deep and cold. It's 3.1 degrees Celsius out there.
Anemone?
So we're out here for another week. We've got a week remaining of this expedition, and we will be spending more time out here in the Coral Sea and some of the shallower coral reefs. We've got some dives planned at Diamond Islets, also called the Tragos Reefs, and Lihau Reef. That'll be near the end of next week. We're spending 2 days here at Osprey Reef. Then we're going to transit back to the Great Barrier Reef margin and have another look at one of these canyons that we're interested in around the vicinity of Cairns, but on the deeper margin of the Great Barrier Reef. And from there we will then head back out into the Coral Sea Marine Park to visit this Diamond Islets and Lihau Reef. That will take us to the end of next week, and after that we've got about a 2 or 3 day transit to Brisbane, and that will be the end of the expedition.
Sadly, it actually went by really fast. Sometimes it's like month-long trips can be, every day at the same thing, but I feel like every day we're finding new things, and I mean, it's very exciting. So.
Yeah, but we're halfway through our expedition, leg 2 of the expedition anyway. Oh, it looks like we've got a wall coming up. So
Yes, there we go.
Okay, big steep wall. Okay.
So before we start climbing up that, he has to move quite a bit because of trouble moving and his headaches. He's coming around and move south.
Yeah, so we're just repositioning the ship. We're nearly 1.5 kilometres down, and we're just getting the Falkor to catch up to the ROV. Big wall, but also, look at what's just to the side of it. This looks again like a big debris field. Imagine a chute of larger boulders. We've crossed that further down, and then this is the smaller pieces jumbled in amongst lots of sand. So, like an avalanche chute. Yeah, very steep. The 3D mapping that we've done over the last day certainly shows that. We've got eel, stalk crinoid. Yeah, there's more of them.
Eric, how's that heading for you? Is it working better?
Wow, that's one big rock face.
We need to come more to port? Yeah. Alright, I'm going to ask
The transect is on top of this, is that right?
Yeah, we'll ascend to the crest of this rock.
Nice, that big bamboo coral.
Are we safe going to that?
hang out for that.
Come back to it. That's okay.
Yeah, it's just that he's spinning. So.
Yeah, the ship's still repositioning itself.
We're actually far enough away to use the lasers to see the length.
Yeah. All right, this is a classic big outcrop. So you can see we've seen a lot of boulder debris, and we've been curious as to where it's come from, and this is quite a big bare face, so I would say what we're looking at further down is probably broken off this large outcrop of rock. Now that is a really long bamboo coral. Bamboo coral?
Yeah.
Yeah.
Yeah, you can see the white skeleton and the protruding polyps. Yeah, we've seen a lot of tall corals in this area, which is probably advantageous in this area where sediment's kind of floating around and burying things quickly.
Yeah, just pick it up so he gets in front of us again.
Wow.
Crawl along this for a little bit. It should only take him a few minutes to get to the safe position.
Oh, jointing in there?
Yeah.
Very steep.
Some joints that are offset. So,
Yeah.
Big sea cucumber over here.
You want to grab a loose fast? This is the fresh face. Yeah. Yeah.
See how it's broken out? It just seems to have fractured, and it's just falling out from the edge.
I gotta run. I gotta run from the ship right now, so.
Run.
Yeah. He's starting to make it dangerous. Alright.
That's right. Ooh, that looks different.
Yeah, sorry. We're running. Yeah.
Chase is on.
He's spinning, and as he spins, it takes me from being behind him to along this ship, which takes my umbilical underneath the ship.
Pulls,
right,
yeah.
where the spinning blades come. Right.
So we're just still doing a reposition here. But the weather conditions are beautiful. You can stand, it's a lovely sunny day, and the seas are absolutely flat. And you can see the coral reef about 3-4 kilometres away from where we are.
Yeah, we've gotten real lucky on this trip. Every day has been perfect. You know.
Yeah, the
flat
weather's
water.
been really great.
Still good. Yeah, it's better now.
Did you want to take out? Yeah, show you the break. Yeah, down the middle. Glasses. Once we settle, Chris, when you're happy, I might find a loose rock to pick up.
We can go a little bit further and try to find a rock.
Yeah.
He's moving to a safer spot for me, and I'm moving in a safer spot for him.
Okay.
260.
Yeah, we're down to 42, yeah. I'm just going to sit down here, we can grab a rock, right?
So the light's low. Oh no.
We're just high up. Sun's yeah. You can go with one of those rocks right there.
Yeah, anything that looks within reach and
It's not going to cause a landslide.
not cause that's right, not going to cause a landslide, and not too big. This will just help us to characterise this rock face with confidence. We collected one way down at the foot of this slope. Good to get another one just to compare. They look identical, but you never know till to get a fresh face to look at.
Scott.
Yeah.
I can have a 3D so we can see where the bottom of the cliff is.
There we go. There's a 3D image coming on view for everybody, just to give you a better sense of where we are. So we're climbing that. You can see that darker brown line up the cliff face there. We're about a third of the way up.
And we're down. Pretty close. See a little encrusting sponge there.
So what's within view and what's within reach?
Have one of these little rocks in front.
Yeah,
Can you zoom out?
All the way.
About this one?
Yep.
What? The big one?
Oh no.
I don't think you can get that one.
Oh no.
I don't think I can fly if you got that. Yeah.
Maybe down on the.
Yeah, one of those ones in the middle.
Yeah, one of those smaller ones.
Yeah, they don't need to be massive.
Ben is itching to grab something big.
Pretty good grabs and shit and didn't do anything.
Okay, yeah. See what we've got there that will come with us.
You did a good job with the alternative halfies, I'll give you that.
And that's it. Yeah, the one below might be loose.
I meant the little one.
The one right here.
It's a little one.
Yeah.
Yeah. Even
It's a pebble.
oh, that's good. Let's get that one now. The moving.
Yeah.
Flat is good. easier to pack. Easier to.
Ben.
Alright, that was moving.
What do we got?
Keep going. Alright.
Yeah.
Yeah, it's in there. It's small. It's in there.
That's a good catch. No, that's perfect.
What do you want?
Thank you.
Two, probably.
No, that'll do. Unless.
Oh, that's what I meant.
Yep. Take that. 2B. Oh, sorry.
The second one.
The second box to put it in. Confusion reigns. Great.
Copy.
Where's this going?
One big.
Drop.
Stuck to the claw. Here we go. Thanks, Ben. That's great. Thank you. We'll continue on up. We need to get up to our 500 metre transect to give ourselves the time we need on that.
You head back over there now. He's much safer.
The ship's, that's fantastic. Okay, that was a good use of the time while the ship positioned.
Exactly. What's this brown?
Yeah, a bit of oxidation, staining and that in some of the rock faces. Not a lot, but it's still there. It's probably where the manganese crust hasn't grown on the rock face, just below that sand layer. Rightio.
I have to go further east. Yeah, definitely. Yeah, you get that turning after you get on that.
Do we have to get back to the start of the transect for the 500, or can we do it offset?
We can do an offset
We
up,
haven't even gotten up to that point yet. So we can,
But can we go straight?
swing around.
Parallel to it and start?
yes, we don't have to go all the way back now.
Yeah, I'm happy with that.
Alright,
Yeah.
so you want me to just start
Just-
right here and go?
Just go up to the.
Well, go to the start, but offset.
Offset it? Alright.
that.
Yeah, that's that.
Alright, so we're gonna.
That's
we're
fine.
gonna go all the way up here. We're gonna go right. Start. We're gonna go to right here. Alright. So we can just bring him up.
Yeah.
It's like we've done this before.
Yeah, we could even start it earlier if you want to.
That's true.
We're gonna get hit and leave it.
10 to 4, 10 to 5.
Further east. East. Yeah. From along that.
Can we judge it from?
Yeah, just take it away.
Hey Eric,
Yeah.
we're gonna cut out north, so we're just gonna be northeast, northeast.
All right, northeast. Thanks.
Are you worried that we're not going to finish in time?
Yeah, we lost an hour at the start of the day, so we'll do our best. We can go a bit longer. Haven't seen J-Rod to.
Alright, so we can start the actual transect, so.
Okay.
The main thing is to get the 500 metre light transect in, then
Yeah.
if the profile finishes early, that's okay. Okay.
With a Gorgon's head, octocoral.
Yep. Yep.
Coming up. Oh yeah.
No good.
I've got some rock.
Not expecting that quite as steep as around the actual one, but.
That'll do.
Oh. Let's look up beyond that.
So very much a debris slope here. All these very coarse, angular fragments of the rock which have broken off over time, from this rock face we're just coming up to now. And haven't travelled far, that angularity is retained, so that's the classic indicator of locally derived material. It hasn't been transported far at all. And here we go into that rock face. Oh, this is neat.
Glass sponge. Some encrusting sponge on the rock.
Mr. Cody.
A very small Gorgon's head up to coral.
Yeah, you're starting to get a sense of bedding as well in these rocks. In this case, looking up and down in the image, more or less vertically, close to it, very steeply dipping beds.
We're coming up, we're going to do the transect northeast. We have original, he says, time, sorry.
At least that's the...
Eric had to switch his head.
Whether they're beds or fractures.
So, sorry, the transect.
What do you think about that?
500, but we're just going to do it on the parallel line we're on right now. So just hit the
Yeah.
It'll start to start, but it's going to be awesome.
Thanks, Adam. Yep.
Start pretty soon.
Sounds good. All right, guys. Thank you.
Thank you.
Proud of you.
Ben did really good. He got me a nice coral.
Did he?
Oh yeah, nice.
Did?
Oh yeah. No.
SuBastian.
Smaller one, but he did good with that one as well.
And a couple of good rocks too.
And he got some rocks.
Did you want a nice wide view like this?
For now, yeah.
Yeah, this is good. This is...
Thanks for asking.
And what speed were you guys going?
Fast for now until we get to the transect. We're trying to get there.
Oh, I see. Okay.
Yeah. Keep our eyes peeled for any bits of colour and sponginess, glassiness on the seabed.
I'm not seeing any bedding or strata in this.
Well, I'm seeing structure.
There's faulting.
There's faulting
going on, but
yeah.
I'm not seeing any kind of exposed strata. I mean, like yesterday's.
No, in total contrast to that. I agree, Rob. Yeah. But when you zoom into the, a single block, it's massive in structure, isn't it?
Yeah, so I mean, we're talking about it from the perspective of a geologist. When we're looking at a big face like this, if you're standing at the wall of a quarry, you can see a face, and you look for things like internal layering, and that would indicate a sedimentary rock.
Yeah, we're not seeing that yet.
But we're not really seeing this. We're seeing a lot of fracturing, what you might call massive. Are there layers here, or are we just looking at it because it's all dipped and it's dipping and it's not horizontal. It's hard to tell. Of course, having a sample will give us a clue when we get it back on board. But right now, I'm not seeing any kind of obvious layering. And the boulders have been very, it's all chaotic and, large blocks, smaller pieces, all different sizes, some slightly smooth, others very rough, angled. Unless...
you can
maybe.
There are large-scale planes.
Yeah,
whether they're faults and fractures is the...
So if you're looking at that, if you imagine it's dipping down to the left, that might be the layering that we're seeing here. And that it's fracturing along that weakened surface.
Yeah, look, right now on that right-hand half of it, that's a failure plane.
Yeah, okay. So that might be a bedding plane there, and then this might be another one.
Yeah.
So dipping, what we say, dipping steeply into the seafloor. And that could be the internal layering, and the whole thing is being folded and tilted sideways. I mean, that's quite possible. It certainly has a long history, this Queensland plateau. So, there could have been folding that's tilted the strata, to dip very steeply into the left here. Okay. So if we take that, so this could be a bedding plane here right in front of us on the right-hand side.
Yep.
Then another. Okay, this is making a little more sense now.
Yeah.
That's just a boulder that's fallen down in the middle. So we've got one bedding. Yeah, it's just steeply dipping into the earth here.
There's no apparent grain size. If it's sedimentary, there's no grain size contrast. It's all just probably another finer grained mud, sand, mud, siltstone perhaps. We'll see when we get our samples on deck. Got to break them first.
So the older history of this is that it wasn't until around 20 million years ago did the plate, the Australian plate that this is sitting on, was first arrived in the tropical warmer waters. So at the very earliest time when any calcium carbonate could have formed, and we're not just talking about coral, but it could have been like bryozoans and things like this, was about 20 million years ago. That's the earliest time that any kind of limestone could have formed. I mean, there's some real simple tests we can do with the rock that we brought back. We can just even expose the surface and put a bit of vinegar on it. If it's carbonate, it should fizz.
So we don't have any acid on board, do we?
Yeah. Other thing we can do, we can just rock saw it, cut it in half, and we've got hand lenses here and some microscopes. We can actually look at the grains in our lens, and it can tell us a lot just looking at it. Still going up though.
Yeah, we've got a fair way to go up. This is great.
Bamboo, just some skeleton bamboo coral here on the right. It's like another feather star just up here in the middle. Yeah. So our map, our 3D map that we generated this morning there, Scott, was around about a 200 metre high cliff.
Yeah,
and that's certainly how it feels, doesn't it? But it's one thing to map it and look at it in kind of a 3D computer thing. It's another to see it in reality, I'm seeing still more of these steep dipping planes
it's pretty
here.
consistent. Yeah. And being so steep, that's helped all those failures to occur when the rock finally gives way.
Yeah, it just slides straight off the bedding plane or the fault plane.
Yeah.
It's another Anthomastus soft coral just popping into view. And I think that's a golden coral. Ooh, just passing over a nice sea cucumber. We've seen a few of those on this dive already. Because there isn't a lot to actually feed on. I mean, there isn't a lot of sediment. It's just pooling in the flatter areas, but for the most part, this is just a great rock wall.
Yeah.
Still seeing.
Yep.
Seems to be pretty consistent, isn't it?
Dipping down slope, dipping west.
So I think that's a good golden coral, Chrysogorgia.
So it's a thrust block. Ooh, look at that. Yeah.
So we're about 3/4 of the way up this cliff now. I'm just looking at the 3D model, which can track the ROV along it. Yep, it's still very steep. And once we pop out on top, we're going to start our 500 metre transect. Some more octocorals over on the left. Nice little cluster of them.
Sea cucumber.
Of course, there is plenty of life. If we were a lot closer to the rock, you'd see that there's life everywhere. I can see lots of brittle stars, lots of small sponges, soft corals. It's just that we only have the occasional larger coral colony. Boy, what's this for a chasm? Gee, look down there. Hope you don't have fear of heights.
It's just peek around there, Adam. If we can have a look, mate.
Wow,
and see what we're dealing with here. That's pretty cool.
Gee whiz.
The rock climbers were like that, wouldn't
Yeah, you could abseil down that. Boy, that just seems to go on forever. Get dizzy looking down that. That's amazing. Wow.
Oh, groovy.
It's like flying off the edge of a building.
Everyone got vertigo?
Oh my god, look at the size of it. Look at that vertical face. Wow. It's like a big underwater mountain. Okay, so you think that block's just fallen out? That's what it looks like, doesn't it? It's just pulled away from the wall. It's like a big tower. Oh, that's nice. Yeah.
Oh, a little mini garden there.
Yeah.
There you go.
A
Just
lot
a little gathering.
Yeah, stalk crinoids. There's two different kinds. You've got this sort of more sparse yellowy one, and the shorter arms and very red. So we're seeing two different kinds of sea lilies, stalk crinoids. I can see some, looks like some anthomastus soft corals down the bottom.
Andy Carroll's online on Facebook saying living on the edge.
Yeah, it really is, isn't it? Yeah, it's like a helicopter view. Just
oh, that's one.
Amazing.
That is great flying too. Bloody quite easy.
Okay, looking up the wall, still going up.
Do you want me to look down while we're going up, or can you put it straight on?
Yeah, this is good. This is working for us.
I'll give it a view to the right or left every once in a while.
Thanks, Adam.
That's a good thing to do.
Just get perspective of how big it actually is.
Yeah.
So we're just past 1,300 metres. We're going to climb up to around about 1,200 metres. Be at the top of this this cliff, and we'll start our 500 metre standard transect.
Very soon, in fact. We're going to run parallel. I don't know if whatever we call that. It's going to run parallel to our track, rather than rejoin it.
Okay.
What do you think, Alex?
Yeah, they look pretty much parallel.
Pretty much line up with that start point.
Got about 12 meters to get onto the start point.
How many? 12.
Yep.
Okay, we'll start it.
Yeah, do you want to start it here or do you want to start it at a certain depth?
No, here's fine. Yeah.
Okay.
12 metres or 10. Yeah, go for it.
Okay, so just close to the wall, coming up slow.
Yeah.
Yeah.
Start transect.
Start transect.
So as well as being a discovery expedition, part of the aim is to do hypothesis testing, and one way we do this is by having our 500 metre transects where we try and we pick a point, we run for 500 metres at a set height, set speed, and of course we've got still images every 5 seconds, and we are analysing each one of those still photos for all the marine life that we can see within it. So the idea is not to stop unless we see a Dumbo octopus or a nautilus.
They're all asleep today.
Yeah, it better be a good one.
It better be a good one.
Yeah, and of course we're comparing each one of those transects across every one of the sites that we visited, both in the canyons that were on the Great Barrier Reef margin where we were in the last few days, and also out here in the deep coral sea. See, it looks like little branches here, Scott, like like coral skeleton.
Yeah.
So just in amongst the rocks.
Oh, okay, yeah.
Yeah, you see these sort of long things.
Filament.
Yeah. So as they die, they're probably just falling down.
Yeah.
Like just in here.
That's great, Adam. Continue on that.
Just continue.
Continue on that. Yep. That line and speed. That's good.
Yeah. Okay. This is skeletal material, coral skeletal, and it's stained, so it's got that dark colouring on it.
Yeah.
I'm seeing lots of sponges though. Lots of these white sponges on the rock, glass sponges.
That's nice.
Yeah, nice stalk crinoid coming into view. Just fly over the top of that. Looks like
mini debris field.
Yeah, it looks like it's cemented together. Actually, a lot of that is coral pieces. To see all the branches.
Got to be coming from higher up.
Yeah. Actually, look at this.
Shoot.
And it's intermingled with the rocks. Yeah. Okay. So there's a lot of coral pieces come down here. You know, some mass death event, or is it just normal coral rubble as it dies? It's concentrated into one little shoot.
How far off the seafloor would we be?
It's hard to tell because the filter's kind of been ROV, so right now like a 2.4.
Okay.
But sometimes when the wall is not showing anything, it's
Yeah.
just straight down.
Yeah, we're roughly about 2, maybe 3 meters just off the seabed here, because it's tricky being in such steep terrain, but that's roughly what how we're flying on this transect.
If you look at my upper right screen, it says alt.
Oh, yeah.
And see how it kind of gets weird for you is when it backed up a little bit.
Yeah.
Now it's 8.9 because it's shooting kind of below the cliff.
Yeah. Nice urchin on the clinging to that wall, and we've seen quite a few of them on the soft sediment, but they're absolutely vertical rock face. And we've got a bit of coral popping out on the side here.
Oh, good.
Yeah, Anthomastus. This is this lovely pinky red coral. Oh, there's quite a bit here. Yeah, we've got crinoids, commatulate crinoids, different kinds of soft corals, some hard corals. Look in the distance, there's a few colonies way off there.
Yeah, getting a bit more.
So if any of these die, they would roll down slope into that chute, perhaps, that we just went over.
Yeah, good point. There's quite a bit in this spot, isn't there?
Yeah, nice. Okay. Nice. Oh yeah, look at the colour of that. This is the Gorgon's head coral.
Definitely a popular habitat.
Yeah, and then we still see some dead. You can see some dead colonies there, just clinging. But yeah, if they got knocked off, they would all end up in that big chute, wouldn't they? They would just roll down slope. So you think over a long period of time will just naturally accumulate within those chutes. Oh, lovely! Look at this.
Nice little garden.
Oh yeah.
Top. Oh, nice.
Beautiful. Oh yeah.
This is what I've.
So these are
for here.
These are commatulate crinoids. These are feather stars, but they've got short legs. They're mobile. In fact, they can also swim.
Just let me know if you want to stop.
No, we'll keep going.
We're on our
transit mode.
Yeah. Unfortunately, that would have been a nice place to stop.
Steam ahead. Yeah, it'll be more.
Man.
Yeah. We just went over really nice.
Yeah, I'll watch every room.
Yeah.
Stylastrids and
all kinds of things. Yeah. Lots of different feather stars. Yeah, Jeremy's just joined us. We've got TV screens all over the ship, so you can be in the galley or out the back or even in your cabin, and you can just dial into what's coming in on the screen. So, you know, if you're really curious, there's you can always watch. Or it might have been one of those corallimorphs, perhaps.
Maybe.
Yeah, that's a nice one.
That's got a lot of tentacles.
Yeah.
Heads.
Yeah, silicious sponges. I don't know if you saw, Jeremy, there was a chute with lots of these, like the dead debris, like the coral pieces, really just covered the seafloor. So at the base of a very steep cliff, we're thinking it as it dies naturally, it would just roll down and accumulate
Maybe
in
they're
one
growing
spot.
to a certain length, certain height at which the friction is too strong and it just breaks off.
Yeah. So if you look through on the seafloor, you see all this stuff. This is all coral debris. It's got manganese oxide covering on it to give it that dark.
So that's like
look.
what we were dredging up last year.
Yeah.
A lot of this.
Yeah.
Just
Dead fossil material. Yeah.
They're having a go at us on the chat.
Yeah. The list of things we stop for keeps growing. Oh, look at this. This is really interesting. Oh, look at these are really big column.
This
dead
is debris.
This is
Dead hard corals.
yeah.
This is what this is.
Wait, but these
Big branches.
These wouldn't be hard corals.
Yeah, no, it's too deep for them. But really big, thick branches. What's this, Jeremy?
I'd be interested.
Can we?
Pick one up?
Throw
Can
one in
we-
a box.
Yep. Yeah, we'll call that.
This is curious. This is interesting.
No, this is,
Yeah,
this is
we haven't
important.
actually got our push call. Maybe we can.
We did at the bottom.
Okay.
But it'd be too. Look, you've got fallen over.
Look at this.
Collapse.
Big
It's a
colony.
Gorgonian.
Gorgonia.
It looks like.
The size of these. These are really thick branches, though, Jeremy.
So that must mean there's a lot of coral somewhere. There's a source.
Higher up. Yeah. Well,
Yeah.
Well, you.
Tomorrow potentially we'll see it if
Well,
it's not
no, man.
in that middle.
But we're
bit.
we're at 1,200 metres. We're still a fair distance off from the shallow coral reef.
The morphology of that.
I mean,
That looks pretty
they're-
chunky. I mean,
We haven't seen
like-
anything this thick yet.
They're more like trees than corals.
yeah.
I mean, look at the branch. It's not. There's no consistent pattern to the branching. We'll just have to take a look and
Have to take a-
crack one open.
Yeah.
So if we can do your best to
Yeah.
Trying to get a
pick up one of those, Adam. Or,
Big ones.
Yeah. Well, the thing is, the manganese, the oxide buildup will make it much thicker than it actually is. If it's
Yeah, true.
old,
True.
They may
yeah.
not be as big.
okay, well, let's wait and see.
But it's been there a long time because this manganese oxide covers all fossil material.
Oh, it's fragile.
That's okay.
It's cemented.
Very well. It's breaking up.
It's not lying loose. Oh, okay.
That's a
there
good
it is.
piece.
Sorry, not quite one.
Yeah, you can see the white skeleton inside.
So,
That's groovy.
when we dredge these up a lot, we're getting a lot of precious coral. I mean, this is
It's
probably,
fragile too.
God.
It's very brittle.
That's really brittle.
That's interesting.
Okay, so really old, bio-eroded,
Yeah.
with a crust of manganese oxide. Oh, we definitely need it. So, yeah.
It's white.
Sorry, Alex.
So maybe bamboo.
It's just that we haven't seen anything living quite so robust, have we? You know,
Right.
a branch this thick. So
No.
Yeah, that's what I'm saying. This is.
unusual. Yeah.
Very fragile.
Oh, yeah. Okay.
Yeah.
But it's okay, yeah,
That's fine.
Whatever
Okay.
that
We'll take that. We'll take that. That's.
And you can throw it in 1C.
Yeah, 1C.
So, is this precious coral?
Don't know.
We were getting a lot of precious coral like this in the dredge last year.
Yeah. Well, maybe. I don't know. We could take another. Yeah, let's grab another one. Stick it in the same box. Thanks.
Okay, 1B? Yeah. Oh, 1C.
1C would be great.
Are you able to grab the remainder that broke off?
Yeah, that's interesting.
So you just need a piece long enough, Jeremy, that you can get the polyps. Just.
Well, there probably wouldn't be polyps.
I don't think so.
And they would die and shed off. So what we'll have is the
Like sclerites, that's what I mean. You know, like the skeleton sclerites. Enough of those.
Yeah, well, if it is a precious coral, we'll be able to tell by looking at, the color. It looks a bit pink, red. If it's got rings, we'll try to figure out what it is.
Fantastic.
Yeah, because bamboo, that will be obvious. It'll have the internal rings, but I can't quite see that here.
These are
Just so robust.
Yeah. Well, see more.
it's very white.
So precious coral. That.
Well, we have precious coral here on Osprey Reef.
What depths does that?
They're
Within this zone.
It's in this zone. Okay.
Yeah. But when you break it open, it has this pink internal colouring even when it's dead, right? So it's
Right. Okay.
it's quite
This is
obvious.
Fragile
So
stuff.
now what I'm interested in is, are we going to find a field of precious coral somewhere?
Yeah.
Well, yeah. So
And that goes back to protecting habitats. If we find an area that has a specific type of coral that's protected, that's information that will go into protecting,
yeah.
Yeah, that's a great point. Yeah.
So I was involved with the German Deep Down Under expedition, great name, in 2009, and they put an ROV down to about 700 metres here at Osprey Reef, and it was the first record of living precious coral.
Okay.
And it was a
Here at Osprey,
it was a
right?
coral reef site in the journal Coral Reefs. So, but very pretty and living, and I think that was recorded around 600 metres depth.
Okay.
And look, it definitely has this pink coloration.
Yep.
That would be really interesting if this is precious coral, which is genus Corallium.
Oh yeah. So if it's like a sink where like it breaks off and falls, maybe it'd be above and not too far away.
Tune in for the next episode.
Okay, so now we only stopped for double octopus,
Nautilus, fossil coral forest.
isopods, and fossil
You want another
precious
picture?
coral.
That'll do. I think that's great.
Cool.
Okay.
It looks like all that stuff that was underneath it, like right there, it's the same stuff.
Yeah.
It's all the same.
Yeah, that's right, Cody. Yeah.
Yeah, and these are big colonies. Whenever even if they're not as thick as we see, they are tall.
Yeah.
Thank you. On we go.
That's good. All right. So
Resume our
start
transect
Transect. Yep.
for Alex.
We saw that one big piece that was fairly complete, isn't this?
Yeah, that's nice.
So we can look at the branching structure of it from this image and maybe try to match it up with a precious coral that looks like this. But this is clearly,
Wow!
complete
Look at
colony.
it all accumulating up there.
So it's got branches and then
You first said Gorgonian, didn't you? I think when you saw that.
because it looks like a sea fan.
Yeah.
But Gorgonians,
We have
they
lizard?
break.
They're
They're
much more brittle.
that's right. Their skeletons made of
We get
scleros.
Lasers run.
No laser.
Yeah,
we
decent
go.
size. Wow! Look at it all, just jumbled up there.
Just so we have the scale of what the size of this is.
Yeah.
Oh yeah, they're just.
Oh, and they're there. Yeah, okay.
I mean, if you look at how steep the reef is, at
Yeah.
the edge, if you have a cyclone passing through or something, and break stuff off.
Or big. So is this a cusk eel over here on the right? We got our fish people ashore.
Let's get a gravity flow. Yeah, okay.
It's interesting, huh?
All right. So what are these corals up on the fringe there?
Those.
They're quite robust looking.
Yeah. And those look like, are those celastrids maybe? The white
No,
Gorgonian?
no, I don't think Solastra is there. Solastra is found a little bit higher up in the. Here we go. So more pink.
This might be what we collected at the beginning of the dive, which we thought was a soft coral. It had 8 tentacles, but it was hard.
Yeah, okay.
Brittle.
The rock there, did you?
Brittle. So we'll figure out what that is.
Just making sure it's hard.
That's right. That's right.
That was hard too.
That's hard too. Sebastian's got good armouring, I'm sure.
Still lots of coral debris. Oh wow! This is looking
glass
good.
sponge.
Yeah,
Glass
yeah.
sponge.
Oh, lots going on here. Yeah, I mean, and also just look at how much of the dead coral is in between it.
That's intriguing.
Yeah.
It has to be this coral here, Jeremy. I mean, there's a lot of it, and it's pretty robust looking.
Yeah.
That's
but the branches were so thick.
Yeah, yeah.
Yeah, look at this.
Yeah, that's
It's
a
quite.
crazy thing.
Yeah. I'll wait to see bigger colonies that would. I mean, but I guess when they get to a certain size, they break, right? The bigger they get, the more likely they are to break off.
So I think there's just regrowth. They're just smaller versions of what we've been seeing. Okay, well, do we want, to take a sample at some point? One of these lighter coloured ones, and compare the
with the fossil.
with the fossil stuff.
Yeah. Let's look for a good one. There's lots, obviously.
Oh, it's a great shrimp here. There's big long legs. It's a good size too. Yep, dinner plate size.
Look at this one. A big sample. Big bit of colony. It's a lot of it. I guess it was.
Wow,
Is it easy to grab? Okay.
Okay, yeah, that's actually quite a good one. They're fairly common, aren't they? When you look across the landscape, they're the ones that seem to be most. And I think of is it Madrepora? Madrepora? It's got the zigzag. It's got that zigzag look about it. I'm just trying to think my soft coral.
6:30. Is that on deck or
6:30 on deck?
6:30 on deck. Okay, guys, that's the call. So you got.
It's the meat.
And the thing is, the skeleton's
Get a
white,
meeting for
right? So it matches the skeleton color.
So
Which one do you want?
we've got an hour.
The one right below us.
One more hour.
I think it's immediately in front of us.
That one over there by the lasers.
I think that's the one.
It's right in front of us.
Oh yeah,
this
right
one here.
in front of us.
Yeah.
Just reach down. Cool.
Okay, stop the transect. Yep.
Would you be able to zoom in for him? Just hold the.
Zoom in.
Yeah,
Sorry.
if you just follow him over, perfect.
Oh, wrong way. Yep. How's that?
This one right here, right?
Yep. You think it's on a rock that's loose? No.
I'm gonna try to grab that rock on the angle we're at.
That'd be good.
Yeah. It seems like that's it. That's the coral. It's white, but it dies. That's the living, actually. But I think that's part of the living polyps. We should get this one. Go to the lower.
This one looks like it's actually growing on an older branch.
That one with the little pink on it.
We'll go with the, yeah.
Right there. There. Oh, right there. Okay.
Yeah, that one's got tissue, so we could, rather than just the skeleton.
Look, there's a dead colony growing on the rock on a base.
That's interesting.
Yeah.
That's a big base. Must have broken off and it's regrowing.
I got it now. I was just first right on the edge there.
It was too close to you. Yeah.
So you can see this coral's it's alive and half dead potentially. So in the middle we see the living polyps.
Look beyond it though, Jeremy. Just see all of that in between the rock is just coral skeleton.
Yeah, a graveyard.
Just kind of fried.
Oh, I see, because you're going to.
Yeah, you see the way that rock is coming out.
Yeah, so you bump it more into it.
Yeah. As soon as I get it, break it.
So I think this is what we got at the beginning of the dive as well.
You think so?
Yeah, it's hard.
Yeah.
But that piece that broke off, if you can just pick that up, that'd be enough.
How about both those pieces? Is it okay if it's in two?
Yeah.
Yeah.
Sure.
Okay. Maybe that little crime one that's on it?
Um,
Weather star? Just keep it together.
No. Okay, I think we got two different.
You think this is separate from this?
Yeah, I think so. Like the polyp size is completely different. You know, it could be zoanthids that have colonised a dead piece of it.
We'll have both, so we'll be able to separate them if they're separate.
Yeah, they're the same coral. They broke off right at the root.
Yeah.
That's going in on it.
Ooh, well caught.
Nice catch.
Yep.
So we will be able to look at the coral when we have it in the lab, and we'll be able to try to determine whether the pink living tissue is a part of the colony, or if it's something different. And if it's different, we can separate them out.
We're collecting it at 1C.
1C, yeah. Because elsewhere there tends to be a lot. Are they mostly white, or do they have a tint of a color? And the fossil was picked up did have a white skeleton.
Yeah. It is. But it looked very amorphous, didn't it? It didn't look like there was much internal, like internal skeleton. When you crack open a shallow water coral, you can see all the different sclerite internal growth quite well. This looked more amorphous, didn't it?
Yeah.
Yes, the question is, has this dead coral fallen from the reef above? And no, the answer is pretty clear. We're still 4 kilometres away from the actual shallow coral reef, so this coral has died pretty close to where we are now, and we've picked up some of the dead skeleton material, and we've just
really
collected
making it hard for
a live one.
It's okay. Yeah. So the pink one's in A.
But you'll be able to tell the difference.
So this is not shallow water coral, this is deep water coral, and it's probably growing on the rocks around here. We just want to find out what kind of coral it is.
Yep, we'll get to the bottom of it. All right, shall we continue the transect?
That would be nice.
So what do we stop for? Jumbo octopus, nautilus, dead coral gardens, giant isopod.
It's either white. Interesting.
Oh, look here on the right. Now just big branches. Control room. Hey. Yeah, we're looking at off the deck.
On deck at 6:30.
On deck at 6:30. Look at this. All right, thanks, Eric.
The thing is, it's just so much bigger.
We need to.
Really
we should
robust.
see medium-sized corals. We should see things smaller than this, but bigger than what we collected.
Yeah.
If it's the same species.
The size of this, the branching on this one.
Yeah.
I mean, that's one whole colony there.
Yeah.
Keep moving at them. Cheers, mate.
Yeah, don't mind us.
Yeah. Don't mind them, not us talk. And
Okay, you want to just keep going straight line?
keep moving,
Yeah,
please.
thank you.
Yeah, we need to try
Okay, the transect's back on.
Okay.
do our best to get our
I
transit.
will ignore you until you tell me, go over there.
Yeah.
Stop!
Save time out.
Please ignore them.
You guys get so excited, I'm like, well, I gotta give him a picture of it.
If you hear this,
Like, go,
if you hear this is interesting,
This is
just keep
Alex.
going.
That's the.
Yeah,
Alex
say
dropped
pineapple.
ice.
Pineapple.
Yeah.
That's the code word.
Alex gives it to
We have 50 minutes, everyone.
Yeah, we've
Alright,
got
goals to
to
get through this transect.
got to get a wriggle along.
Yeah.
Hey Scott, quick question about the transect. So
Yep.
that wall, we came up about 80 to 100 meters. Are we counting that as part of the transect? That 100-meter line?
Yeah.
You want to, okay.
Yeah,
So
cheers.
I'll change that to 100.
Were you scoring? Sorry, Alex.
Yes.
Oh, octopus! Stop here. No.
And it's not a Dumbo, but it's still an octopus.
Keep going.
What's the?
Oh boy!
It's a big one too.
Sure, let's just get a quick shot of it.
Give me Connor's. Yeah. Okay.
That's a pretty cool one. That's a big one.
Oh, beauty. Look at that.
That's good.
Oh, yeah. And
Oh,
it's quiet too.
That's
Okay, look for eggs. And I think there's eggs. Are these eggs underneath? These little...
Those are the suckers. Huh?
No, these are eggs. These are octopus eggs. This would be a female, right? So they plant their eggs on the underside of the rock.
Oh, look at that.
Yeah, oops, sorry. There we go. Someone else is in charge. Yeah, so this is a female, and she would be guarding her eggs against predators. And after the little baby octopus come out, she pretty well dies. That's the life of an octopus.
Wow.
Yeah, and it's very quiet.
So
It's
it's safe.
not moving. Not moving. If I got that right.
Yeah.
There we go. Whoop, there we go. Let's just zoom in on. Come left a little bit. Okay, you can see the eggs, and in fact, the little baby octopus, you might be able to make them out. These are the eyes, the little dark dots on the inside. They'll come out as like miniature octopus, of course. So that's fresh.
Wow. Good eye.
Very quiet. It's not moving at all. Like who would have? Here it is at 1,200 meters. Yeah. All right. We are on our transect, so we'll just zoom back out again. That is a great find.
Okay. No more stopping for sure.
Wow.
That's right to leave the room.
Beautiful.
Basically, it's fantastic. Yeah, it's good. But I say
Oh, that's my highlight of the dive.
Yeah, that was amazing. Every dive, you know.
Wow, yeah, it's quite a garden here. So we're on top of that big boulder.
I'm still waiting for the big colonies.
Cheers, let me know.
Where are they from?
Wow, eggs and everything.
It's good that we got a sample of this, you know, because it's clearly dominating this area.
Yeah.
So, do you want to call that 100 meters vertical on that tooth base as part of the transect? Over 500? Or are we doing straight 500?
What's your answer?
So when we started, we came vertically about 100 meters. Do you want to count that as part of your 500 meter transect? Okay. Okay, because now it puts you at like 700 meters for the whole transect. Roughly. If we go to here, so from here plus the 100 meters, we started here, it's about 500.
Yeah.
So about 600 meters.
Yeah, so
That's vertical above
surface.
bench.
Surface.
Okay, so that's full surface. Okay. Yeah, no problem.
It's a good question about the crown of thorns sea star. I don't think it occurred.
Not at these.
Not this deep.
Not these depths. No, definitely not. No. Tom Bridge, who's a collaborator on this expedition, he's not with us physically on board, but he's involved with it. He's done quite a lot of deep water surveys on the Great Barrier Reef. He's seen them down around 40, 50 metres, but not in great outbreak. We haven't seen outbreaks at those depths. They tend to be more concentrated in the shallow waters, say, the top 10, 15 metres. That's not to say they don't go down deep, but we just haven't seen them in big outbreak numbers down in the deeper depths, and they certainly are in these sorts of depths. So crenithorn starfish eat coral, like shallow water coral. Right, we're over a kilometre deep here. It's the coral we're looking at here is cold water coral.
So now it's flat here. We're not seeing any of those fossil corals. So. And are we going up still?
No, we're coming down.
Going down, yeah. You can look at
Dwight's
computer there. You can see
coming down a gentle
off and then back up the next side.
Yeah.
All right. Yep. Just curious as to.
Yeah. So we've come up over the crest of a hill.
Yeah.
Oh, and we've got a stingray or a ray just off. Yep.
we're gonna get on top.
Yep, it's definitely a ray.
We're not stopping.
Deep water ray.
Thanks, Jeremy.
And we'll probably go right over the top of it. Thanks, Adam.
What's that?
There's something floating along just above it.
There's a nautilus.
Headless chicken.
No, headless chicken.
Headless chicken, sorry.
Okay. Oh yeah, it's a sea cucumber. It's just drifting past. We're getting a good look at this ray. It's not very big, maybe 20 centimetres across. Got an interesting pointed snout. That's nice. We have actually seen deep water sharks out here on previous dives.
It's a random block.
Yeah, look how it just concentrates life there.
Yeah, classic.
Lots of anthomastus, soft corals, those mysterious white hard corals. Yeah, like little islands of life in the seascape. But you know, I'm also interested in these strange holes, these funny round shape parallel burrows. I'd really like to know what creates them. So this is this Lebenspuren I've been talking about, German for life traces. Lots of tracks, trails, mounds, burrows, and you know some of the tracks are very obvious. You can figure out what they are, but others are complete mystery. We have seen a lot of these kinds of these sort of circular clusters of holes, but I still don't know what's making them.
I see how I guess it could look like it's going up, doesn't it? It looked like yeah. You're going to be increasing so today
So about another hour on the seafloor?
Might have brought a bit more time. Okay. But for the moment, yeah.
Yeah.
Tomorrow, shallows.
Nice shallow, yeah.
That should be an exciting dive as well.
Yeah, just no Dhugal dive, just a...
Yeah, so the plan tomorrow is we will move closer to Osprey Reef and we'll be diving up the steep flank of Osprey Reef. What starting depth do you think?
Check. We haven't drawn that one
maybe
yet.
Around 600 metres?
Yeah,
maybe 600 metres.
600 to 100.
And we'll go straight up into the mesophotic zone. Opportunity for sampling for Jeremy and the other biologists on board.
Oh yeah, Thomas
Back at 2 p.m.
eager to collect there as well.
Yes, very strange tracks, burrows.
Holes around.
But they're holes. You know, a lot of them have holes at the base, like left by, I don't know, crustacean or a worm, but they have this really distinct parallel sort of patterns.
This is the highest density we've seen, I reckon.
Yeah, I agree.
They're all the same size, right?
Yeah.
And orientation as well. So it's open and we're going through the opening.
So about 20 centimetres across, between 10 and 20 centimetres wide. I mean, that's clearly an isopod burrow, but...
So it's not a complete circle there.
No. Yeah. We've seen them very regularly, but Scott's right, this is probably the densest we've seen yet.
It's not a bad idea. Someone online suggested we take a call.
Yes. So I spoke with Rachel Przyslowski before we came out here, and Rachel's also in the Lebenspuren fan club, and she was on the Ningaloo Canyon expedition that preceded the Falkor's trip over to the Great Barrier Reef. and they did the same thing. They put down push cores into these burrows and came back with nothing. There was nothing inside it.
Biota.
There was nothing really obvious that they could pin it down to.
It's very cool.
Eel coming in on the left.
Yeah.
Soft coral.
Yeah, okay. Some kind of crab down here. Squat lobster perhaps.
Halfway there. 250 to go.
Yeah.
Some of them mounded in the middle here, Jeremy. These are almost clumped, you know, like a low mound.
So are they picking out of the holes and putting them in the middle?
When you look down on them, they're definitely burrows, like they're holes in the sea. It's not like an imprint.
Right.
You know, when I think of sediment like this, I think of worms. I mean, we have seen echiurine worms, but their proboscis comes out on the seafloor. But no, I'm really curious. This is the most common imprint on this track that we see out in the Coral Sea, and I still can't even think what they might be.
Yeah, they're dead right. It's the most common.
Got to see it in action.
It's crazy. I mean, there's nothing to give a hint at all. At least the isopods were there occasionally.
Well, you know, we see the sea cucumbers, and they leave these obvious trails, and we see the occasional sea cucumber, so we can, you know, match the track to the animal. But this is just something.
Urchins we saw, that was active.
Yeah, that's right. They leave that really bulldozed track on the sea, quite wide.
Could just leave the ROV down overnight.
We could.
At the beginning of this voyage, we talked about strapping a dead fish to the front of SuBastian and just seeing what. A bit of chicken, and seeing what came along. It'd be really interesting, because this is how BRUVs work, baited remote underwater video systems, and they've been used extensively here at Osprey Reef to catch nautilus. And we have Dean Miller on board, and we'll see if we can get Dean to have a chat about this. But he was involved with nautilus trapping, and they would use pieces of chicken to attract the nautilus, and they would capture them in the traps and bring them up to the surface and do a whole lot of tagging, and then release them the next night. So he's got some good stories to tell about that. And Justin Marshall, who's a professor of vision at Queensland University, put a baited remote lander down here at Osprey Reef. I think it was just a large piece of fish. And they had these infrared lights, you know, so it wasn't white light, but then they left them there for several hours. and he's got some great vision of a really large 6-gill shark coming out of the gloom and sniffing around the video here at the base of Osprey Reef in these deeper waters. And they're a really strange looking shark. Big blunt head, large eyes, very, very slow swimming. So there's clearly, some large predatory animals around, but maybe we don't see them because we're just moving through the landscape so quickly.
And I think the ROV scares away large sharks because when we scuba dive, we see them when we get down 30 meters. You see them out there. We're facing the reef. We're not looking away. There could be sharks all behind us.
That's right.
But we also have cameras behind us. So I think they stay far away from this large, loud sounding.
Yeah, fair enough.
The Nautilus though are different though. They're not. We don't scare them away. Every time we're around Nautilus, they usually come to us.
Maybe because we scare away the sharks.
Yeah, we put a GoPro down on a stick one time, and you can hear the sound the ROV makes through the GoPro that was picking up in the water. It's very loud, so we suspect that a lot of the things that goes down there we scare away.
Factors in our navigation.
Acoustic disturbance.
And the bright lights, all these things are down there. So what is this freight train coming through here?
Okay.
At least small things can't swim the reef, but shark can.
Could you just put the sit cam in as an inset so we can? This is quite a good view of these burrows, just to show people that what we're talking about, the patterns. So Viet's just brought the inset is the sit cam. This is the wide angle view, and so we're basically looking straight down, and you can see the burrows in these strange tracks on the seafloor. Because the science camera is looking obliquely, you don't always get that, the picture of what it looks like. So you can see these. The burrows they're parallel, and they're anywhere between 4 and 6 burrows either side of each one of those clusters.
And they're slightly tilted at one end to make it look like it's like a horseshoe. But really, they're just 2 lines that are perpendicular and maybe just slightly offset.
Yeah.
Or inward.
All right, we're running out of sand here. Thanks, Viet.
We're coming up against the next part of the incline. Hill number 2.
Because we're doing our transect, we're not going to change the view of the main camera.
Yeah, thanks, John. We can drop that inset out. You can remove the inset now.
Okay, thanks man. So that's on deck at 7. On deck at 7, so we'll go till 6.
Okay.
Thank you very much.
That's one of the anemone crab. Crab anemone. There's an eel in the centre. It's still pretty cold down here. It's 3.5 degrees Celsius. See that kind of bedding plan again. Debris, big boulder field. You can see that's just fallen out of that.
Yep. That's good. It looks so weird because the way the camera was pitched down, it looked like we were coming like this the whole time.
1 more hour now. It's good.
Yeah, there's been lots of surprises on this.
It's been a good one.
I think the highlight has been that big cliff. Looking down over the edge of that was pretty good. But the octopus was quite special. It's very coarse. Looks like the sediment here looks a bit coarse. I wonder what's doing that.
Glass sponge.
Oh, that's a sponge.
It's glass sponge.
Yeah, one of these elongate glass sponges. That's a beauty.
It's huge.
Yeah. Like they call it the Venus sponge. So one of our collaborators is Merrick Eakins from the Museum of Queensland. He's a deep water sponge expert, and he'll be able to identify that really clearly. Yep, we're seeing lots of people saying wedding sponge. Yes, so this is this tradition in Japan that they give each other these beautiful long like elongate vase sponges, and because there's I forget the story. It's something to do with how they are committed to each other in their marriage, and so it's a wedding gift.
Okay. Right.
There's 2 shrimp that go into it. They can't put back out and they make life.
What's inside it?
2 shrimp.
2 shrimp. Okay. Yep. From Cody, he's saying that 2 shrimp live inside it. I assume a male-female pair of shrimp, and they live entirely of their lives within the internal parts of the sponge, and so that's the symbology of the gift for marriage in Japan.
The offspring are small enough to escape. And they stay there for the rest of their lives. Yeah.
We won't need an hour, but it's just good. Pressure's off.
Yeah, so we've got about another hour on the seafloor here. It's 5 o'clock in the afternoon. Still very nice weather conditions outside. But we'll go as long as we can with daylight here. There's a golden coral just passing over. Some fairly large colonies popping out here. Skeletal material on the seafloor. Nice squat lobster. Yeah, thanks, Jason. A Venus flower basket sponge.
Zone heart?
how the corals are growing up at fracture.
Cracks,
Yeah. They're just finding something about the that line in the rock, and they're all attached to it. That is really interesting. Oh wow! Look at this, a fringe.
And then on the crest.
Yeah.
That's a nice.
That is a very pretty view, isn't it?
Isn't it?
Yeah. But maybe on the the sharp corners of it, because this is getting
coming up.
more food, more exposed to the water.
Yeah, absolutely.
Water flow. I mean, they're relying entirely on the water flow past it for their food. And down on this rock, you can see.
It makes sense.
Yeah, really interesting how on the one that rocked the bottom corner there.
I know we're not supposed to change direction for the transect, but if I turn just like 10 or 15 degrees to port, I'll get more of a straight on view, so you'll see more as opposed to that kind of like strafing up the side of the hill. Is that okay?
Alex?
Just
I'm happy. Yeah, for that. As long as the imagery is good. Yep.
Yeah, so we're still on our 500 metre transect, although we've stopped a fair bit along the way. And we're seeing more of these dead coral colonies. Lots of it, all mixed up with the rock boulders. Really thick branches, aren't they?
Yeah, but they're bulky.
So Emily's asked if it's a dry boat. No, Falkor is not. Everyone can have a beer or a wine with their meal at the end of the day, but no more than 2 drinks each, and not before going on watch. So very much in moderation, but yes, people can have a drink of alcohol with their at the end of the day. Wow, the size of those branches. It's just interesting. I mean, we've got lots of corals, but nothing quite as thick as that.
Yeah, that's right. This is nice.
Okay. So one big one coming up here, watch this.
Very pretty brittle start. Oh, love it. Look at this. Lots of little sponges. Oh, what do we got here?
Wow, this is really kicking in.
It's a tree of crinoids, feather stars grabbing onto something. I don't know what it is. It looks like it's broken off.
Yeah.
1, 2, 3, 4, 5, 6. 6 big commatulid crinoids. They can swim, so if you disturb them, they can jump into the water column and actually have this beautiful, graceful swimming motion. Oh, wow. Lots of life here. Golden corals.
Turn again and go up this face now.
Okay, thank you. Yeah. Got a big glass sponge in the background there. Even sea cucumbers getting in on the action. I mean, it's hardly any sand, but they're still making use of it.
Brown.
Yeah, they look sponge-like, don't they? The white ones are really obvious. They're glass sponges. Oh, look at all the dead branches on the right.
Yeah.
They're clearly just breaking off and tumbling down. Big sponge over there.
Wow.
Wow, I mean, the golden coral. Lovely. big glass sponge on the right. Sea cucumbers as well. There's a lot going on in this wall. That's lovely. Pretty amazing, isn't it?
This has proven itself as a good habitat. We thought about that with this dive site, testing the hypothesis that this major block or ridge, if we might call it, is potential habitat. Let's tick that box big time.
Yeah. Yeah, look at the ground. It's a really big mix of rock debris and coral.
A lot more debris.
Yeah, lots of debris on the right, isn't there? In fact, most of that is just coral debris.
Yeah, fascinating.
Wow, that's a big accumulation, isn't it? But just the size of the branches. There's quite a few layers in amongst all that.
And then he overlaid it on our thing here. He's like flying by. Just decorated that clamshell. It's like this. You can zoom right in on a clamshell. It's perfect square.
Are we seeing that that steeply tipping strata again there, Scott?
Yes.
It could be going like almost 80 degrees down into the seafloor.
There we go. Another little
that is
little
nice.
high point for
Look at that.
Congregate on.
Yeah,
nice.
Big sponge. Look at the internal structure of that. Yeah. This is fantastic. Have you been watching?
Yeah. So you reckon these are not thylastrids?
No, I don't think they are. Okay. Yeah, we find thylastrids here, but higher up, closer to sort of around 300-400 meters. We find thylastrids. We haven't seen any at this depth before.
I'm sort of thinking it's a precious coral.
Could be. Yeah.
I was just looking. I mean, obviously, it won't look exactly like this.
Same colour.
Color and then fading to white.
Yeah, we're just looking at we've got Jeremy with us. He's pulled out a coral identification book. We're just flipping through his pages here, trying to identify some of these corals.
I mean, yeah, these books don't have nearly enough to identify what it truly is, but sometimes they can nudge you in the right direction.
Yeah.
Yeah.
Whoa, there's a cliff.
Not far. We're doing well. We'll finish before we need to.
Is the transect on still?
Yeah.
Or is it over?
Yeah, no, we're on.
That's right.
But I think if that detour we came up that face and around, you're probably fairly close to 500 right
Yeah.
now.
Yeah.
It's got about 20 meters to go.
Right.
Yep. Yeah, see how the corals fringe the edges?
Perfect situation, perfect position to get upwelling.
research.
So this really large hill that we've been going up, this is just one of many that are around the base of Osprey Reef, and it was part of the reason why we wanted to look at this one, because when you look at Osprey Reef in 3D, you've got the shallow reef area, you've got the steep flank walls, and then kind of out from the edge there are these platforms, and sitting on these platforms are these really large hills. And we thought it would be really important to try and understand what they are, what they're made of, and of course what marine life is there. But now that we've seen it, it's pretty spectacular. We can scale up. We can kind of scale up the area of habitat that these things cover, and then we can, really start to understand more broadly what's living in the deeper parts of the Coral Sea Marine Park. Big branches over here, and
still there.
Yeah. Oh, what have we got here? It looks big. Yeah, possibly a bamboo coral. Yeah. Get a little bit closer there, Adam. You might be able to see the nodes.
Rock off to my left here, then I'll just have to turn here a little bit.
Yeah. So,
Want to take a look at this, or just go to the next? Just finish that transect.
Finish the transect. Yeah.
Yeah. See how it's jutting out.
Yes.
Stay off the wall.
Sure. Yeah. Yeah, we're a bit close to a steep wall here, so we'll just have to move out a bit. Yeah, I'd say that's it is. You can see it quite clear. It's a bamboo coral. One of those really long ones, maybe 3 meters long. Look down slope, Sky. Do you see all the coral skeletons just lying at the base? And right in front of us? See how it's all matted together? Oh, yeah, look at this.
Patch of it there.
Yeah. It's just, it's accumulated over years and years and just completely matting the seafloor here. I mean, how thick that is.
Yeah. Neat.
No, the SuBastian doesn't run on battery. I think, what's the voltage we put down? 4,220 volts? 4,200 volts, and that's to drive all the cameras, hydraulics, lights. So it all goes down through an umbilical cord with fibre optic vision coming back to us. Yeah, so it's a pretty high-tech equipment. Could we just come over to the right? Thanks, Adam. There's quite a lot growing here.
I would probably say that's over your 500.
Yeah, we're done.
We couldn't do a straight line because we've been off in deep water there. So.
Thank you. We'll end at 500, so we'll just continue on and explore.
So we'll just have a look around here.
Look around.
Plenty to see.
Might get over the top.
Grab some more of that stuff.
Yeah, because it's very interesting. Lots of.
Can I grab this stuff here?
Yeah, if it's in a good spot. Sure.
Would you like me to go into a bigger biobox 2 with one of these?
Steph, biobox 2.
Totally empty.
Totally empty, so put it in there.
These are pretty big pieces.
Yeah. Yeah, look at the size of this colony here. We just parked Sebastian on top of it. Whoa,
So
that's a big piece. Might have to break that one up.
You know, just our luck, as you said that, it's not gonna break.
It's not going to break now, is it? No.
So you think this might be a whole lot of branches that are just fused together?
I think it was cemented together, but yeah.
It may be not that one.
Yeah, just a bit big. There's one that's loose on top over on the left here. Yeah, that might be a bit easier. There you go.
Might have to knock a bit of that off.
So to go into this one?
Yeah.
Okay.
Two's good.
Okay, box.
Thanks, gents. That's good.
So you want to like maybe crest the hill here and then like kind of up start and just run up the top of it?
Yeah, that's fine.
We've got. We can have a bit of a wander around now. Oh, there's a fish down here. Is that one a cusk eel? It's one of those big solid eels. Maybe fish people ashore can tell us what we're looking at here. Big eyes. Are they really? Yeah, it's definitely got a dorsal fin. another big bamboo coral here on the right, and yeah, look at all the debris at the base of that slope. It's just some of those branches. I mean, they're amazing how thick they are. Oh yeah, check out the sea star in the middle here. Can we just have a close look at the sea star there? Yeah, let's have a just wrong way. There we go. What's the rock? So, yeah. I'll go right in and see what we can see. It's one of the biggest sea stars we've seen on this dive.
Yeah, it's really brilliant detail there.
Yeah. Yeah, there's a lot of going on in the rocks, aren't there? Lots of small sponges, possibly. It's like stylastrid corals up on the right. Thanks, Adam.
Yep.
You want to jump in?
Sure.
Yep.
Ooh, nice stuff. What's that? It's been hard to look away. Where the hell are we? We're just cresting the hill, kind of. Ooh, wow, what's going on here?
What's that?
That's a distant view down the bottom of it. Okay. Oh, another face.
Oh, is this one of these big vase sponges?
Oh, maybe.
On the left? Yeah, okay.
Is this the end of the transect here?
We've done transecting, we're exploring.
We're exploring.
Exploring, and we're just collecting.
All right.
What did we just grab? Big slab of
big
the
fossil
coral.
Really
so this hard brittle coral we've seen the entire dive.
Right.
Still not sure exactly what it is. This one looks green, right? But I was,
yellowy green.
I was doing some research. So the stylastrids and the precious corals are very similar looking. The main difference, the way you can tell the difference is the stylastrids will have pores, while the precious corals will be, the surface will be smooth, 100% smooth. And the depth range of stylastrids, usually 200 to 400 meters, but they can occur as far as 3,000 meters. In New Zealand, they can be found deeper.
And in Antarctica, down at 2,000 meters. So
right.
I just wrote a paper about them.
Oh, okay. So this could be, if they're stelastrids, a pretty decent range expansion, bathymetric expansion in this region. And we have samples, so we'll be able to figure it out. So what do you think these are?
Sorry.
The corals, the hard corals.
Yeah, I don't know that.
We'll figure it out. There's lots of holes in the rock wall.
Yeah.
So encrusting sponges,
Lots
bases.
of sponges. Yeah. I'd
bases.
say yellow and those white ones. Lots of sponges.
Yeah.
It's very pitted, isn't it?
Ooh, look at that guy. That's a big one. They're getting bigger. So at the top of this cliff, we have much larger. Oh wait, no, this is different. This, we should look at this.
Okay.
I'll zoom in.
I'll just sit down in front of it.
Yeah. Okay. Okay. Yeah, it looks like it's got 8 tentacles. So my first guess is octocoral. Or a bamboo coral. Yeah, bamboo.
It's got lobsters all over it.
Yeah.
Back off.
Okay, we'll get a nice image here. Let's zoom in on this guy.
This looks like golden coral to me.
Yeah, right.
Yeah.
Yep.
Yeah.
Beautiful.
Yeah, it's a golden coral.
Mm.
That's a great photo. Yeah.
Oops. Look at that gold.
Yeah.
Wow. That's a money shot.
Wow.
Postcard.
Yeah. Beautiful.
Fantastic.
Coral Sea. Postcard from the Coral Sea.
What's this in the background?
See that? Maybe a polyp that fell off. That was a shrimp.
Yeah.
Is he reaching out to say hello?
Smack. No.
Just stretching backwards.
A bit like the Sebastian's arms.
Yeah. Trying to replicate.
So what did you do?
So with the
Nice
lasers.
big colony of big golden coral.
So let's see, we're talking about 10, 20, 30, 40. So it's over a meter height. It's wide. Nice.
Very pretty. Okay. Take a look off the top.
Nah, that's okay.
No, we know what it is.
No, we're good. You're eager to get a big sample, aren't you?
Cody wants to example.
Alright. I don't know anyone working on golden corals. Although I may get a message later saying, why didn't you collect that coral? It's perfect.
We'll send you a message saying, we told you so.
Oh yeah. I'll let you know. It's a really awesome dive.
Okay. Yeah.
Really awesome.
You do all the sampling. The sampling of the golden coral.
So we're seeing this one species of maybe salastrid, maybe. We'll let you know tomorrow. Remind us to tell you. Yeah, we'll look at it after the dive.
I don't think salastrid. Personally, it just doesn't look delicate enough.
Well, the salastrid are more delicate, right, than the precious coral. But these have, I don't know. There's a halosaur.
Oh, what have we got down here? Isn't that the cusk eel? I think it's a cusk eel.
I wonder where Gemma is right now.
Yeah.
This would be a fun site to dive in, you know?
Yeah.
Swimming through the rock holes. Oh, Gemma.
Yeah, I've been doing a little research on those guys, and the team on shore as well, the Fish Science Army. Bit of discussion about whether it's a deep sea gadiform, so close to the cod family, those sorts of fish, and that includes a group called the Macroeuridae, which also includes lots of those little rat tails that we see. So I think the first one of these bigger ones we saw, we got a really nice look at its lateral line. It's got that classic sort of curve down its body, nice fins. So I think if we can look through the footage of this, that'll be quite a nice ID shot in there.
Great.
Bit of purple coloration on the tail as well.
There was one that had a really distinct dorsal spine sticking up.
Exactly that. Very cool fish. Their eggs actually, they're lipid-rich eggs, pop up to the surface, so they hatch up shallow, and then as they develop, they'll migrate downwards, and then as an adult, they'll be living down here. Lots of sort of sensory ampullae on the underside to help them sift through the silt, foraging and looking for little tasty treats in all this sediment. Big eyes and big heads. Sort of that's where all their good stuff is for helping them live down here. But yeah, really nice. I think they're really pretty. They've got
We've seen about probably three on this.
Yeah.
Not a lot. They've been sitting all by themselves, often in crevasses.
Yeah.
They've been underneath some of these deeper crevasses.
Yeah, really nice. What have you got there?
Good talking to you.
Ah, cool. Purple tailfish could be ready too. Oh, this is great. It's so good having just engaged people on shore chipping in with, what we're seeing here. It's really fun to have these discussions to try and figure out what we've got. Such a rare and amazing opportunity to get to have a look down here, and it's just really great to share it with everyone. Yeah. Yeah, some excellent suggestions coming through. So yeah, we'll get back through the footage and see if we can get a nice clear shot of it, and we'll share that around and get a proper ID.
More of this dead coral coming up. It's a real feature of this dive. Yeah, great clusters.
Oh, look how big
of colonies, dead colonies.
Is this one?
Stained, ferromanganese oxide stained, clearly showing a lot of age.
Yeah.
But there's a fish for you.
There's another. I think either very similar or the same sort of species
look
we've
at
been
this, please,
seeing.
Adam.
You can see some little barbels under its chin.
It's one. This whole area seems to be.
Yeah.
Maybe it's growing along the floor and dying, which allows it to continue to grow.
Right.
Maybe, I don't know.
Clearly become home for a lot of lots of other stuff.
Fish is using it as a
Yeah,
shelter.
yeah.
So you think it's upside down, and we're looking at the base of
Yeah.
the coral?
This is huge. Certainly a reef building coral.
Yeah.
Yeah.
Right, okay.
And you definitely see that kind of thing in the shallows after big storms and swells. There's huge tables or plates that tip upside down.
And these are
So like 1,200 metres.
Yeah.
And you know, the shallow reef is 4 kilometres away. This is
Yeah, this was not from the shallows.
But you could see it's been here a long time because there's a lot of growth on the outside. These branches aren't shaped this way. These are just.
So there are a lot of concretions on them.
Yeah.
A species that doesn't exist anymore.
Yeah, it could be extinct. Some extinct species. Yeah.
That's on to that.
We just haven't seen coral quite as robust as this area.
Morphology hasn't been right.
Generating lots of complex habitat clearly for all sorts of other things.
Like a nest.
For fish and sponges.
Looks like we've lost that fish. It's sort of jumping further out. Look at that. It's just like a tower of skeletal material.
Swim around it.
Let's try to take a nice view here. Wow, just got.
It doesn't feel like something that's characterised by diving around the shallows of Osprey. Like you don't see that kind of formation.
Can we go around this?
There's
Kind
a fish.
of circle up it. Like take a 360 view. Amazing. We're just seeing branches. We're not seeing fixed stems. You know, we're not seeing a stem in it. It's just branches.
Do you see anything that might have been a base at one point? You know, an attachment point.
Unless that is a base.
Yeah. Wow.
See something?
Oh no, we'll just keep circling around it. Just
Having a closer look. It's just a big pile of dead branches.
Wow, it really is huge.
I mean, what if it is this species that's growing, and it just keeps dying and falling, and the base continues sort of living, it grows, it dies. I mean, clearly, this is one species on top of a dead pile of it, maybe itself.
Look at the size.
I mean, obviously, they're way thinner.
Right then.
I mean, it's as thick as my arm in some places.
We'll get some samples. We'll have
Yeah.
you guys on.
Yeah, that's going to be really exciting to look
Like
at.
a little cave over here.
Oh yeah.
So what's at the top should be the youngest, right?
You're right. I mean, this is like its own little reef, isn't it?
Yeah. Unbelievable.
So we saw it in the deep, and we're seeing it again here. So, you know, where's the source?
It is so curious. We haven't seen any live similar
Like really big.
colony.
Coral mountains. Well, so we have coral mounds, you know, and that's usually baffled with baffles mud, like on the Irish margins.
Maybe we'll go up this.
Huge overhang. No.
This is interesting. I mean, we have this fringe of living coral, but in the deep, we haven't actually seen too much of this reef-like structure created, except here.
Yeah.
How far away is the reef edge?
4 kilometres.
Yeah.
A long way.
It's certainly not shallow corals falling.
No. Too far from that.
The fact that it's upside down.
Right.
Means
But is
it's coming from somewhere.
I mean, yeah.
Maybe like a leaf falling from a tree, in a current, like a big structure might just slowly.
Do you think we're going to see a ridiculously large colony that just every 50 years just falls?
But you know, they're so big, you wouldn't expect them to move too far, except on really steep slopes. And these are just on the top of a rock.
And you'd expect them to be broken up. They seem to be big pieces of large colonies.
They're quite localised as well. We don't see them scattered between those patches.
Right, so the source has to be nearby.
Bridge ROV.
If not, right there.
Did you guys
do a little bit of north there?
Yeah.
You see that on the west. Thank you.
Sure. We'll call it in 10 anyway. No, I'll wait till the 6. What's dinner tonight?
I don't know. I'm sure it's delicious.
Yeah. I was not good enough to get one of those chocolate croissants for breakfast as a snack for lunch. So
right.
good.
Was it?
Yeah, that was
good.
It's sweet for breakfast.
Yeah.
With coffee. Something floating down there. Something small. A lot of love for the chocolate croissants on the chat here. Oh,
What do we got? Is that a fish?
no. What's
Or is it a
this?
frog?
Or is this a... Looks like some kind of
Is it a frog
Yeah.
fish?
grumpy looking individual.
Exactly.
Excellent.
Oh, cool.
Oh, wow.
Looks like a frogfish. What's the deep version of a frogfish?
Deep version of a frogfish. Got a suggestion here, a coffin fish, which I think sounds pretty good. It's quite big.
Do we need to white balance, or we?
Yeah, we should, because that's what we did one too.
I'll zoom out.
That is quite big. Probably
Before we get too close on
Yeah,
it,
yeah.
30 centimeters.
You muted it so the lasers couldn't shut down the doors.
10 seconds.
Just gonna re-white balance here so we get a
11 it
clearer
is.
picture.
It was 20 to
Yeah.
What?
All right.
Ah.
So, are we good? I'll zoom in while we. It's big. It's like
Goosefish.
20 centimeters.
Yeah, it is big. At least 20, maybe.
Very sedate.
Maybe 30. Yeah. Clearly a huge mouth. Some kind of ambush predator will
It stands out pretty well.
wait for something to drift by and then gulp it down.
It's got spikes.
Nicely camouflaged, yeah. That pattern all over him.
I mean, I can see him pretty well.
Well, you're not a deep sea shrimp. It does. And look at those fins, almost like
legs.
To
legs.
look
Yeah,
towards the middle of the shot, right? Like that.
it does. Yeah. Ah, this is a lovely shot. That's really cool.
Colours.
Wow. Look at the texture of the, I want to say skin, but it's obviously not. It does, yeah. Elephant skin.
It's got a lure, right?
Yeah.
You're right, yeah. Wow. The things you find down here.
How long ago did that?
What if he yawns right now?
I would love that.
Is this what happens when you flush goldfish down the toilet?
That's awesome. Just awesome.
Can you get closer yet?
These spikes are
Oh, look at the second eye.
pretty gnarly looking.
And just sharpen up the focus just a touch.
Goodness
It should
me.
auto. I'll zoom out in it a little bit. So
Look at the
Look at the lips.
lights of the.
Just hold that for 20 seconds, yeah.
Will that get a good still from the... Yeah.
well, we got both eyes, which is good.
Yeah,
It's nice when
rad.
both eyes are looking right straight ahead of you. If we could. But
You dangle something in front of his face so you can see me.
Yeah, we should...
He's got two little lures.
Yeah.
Yeah.
One tucked away.
We should develop a little fishing rod extension for Sebastian to wave little things in
Come out
front of
to
him.
full fish. Beautiful. Give that another 10.
So what are we calling this guy? What's the name?
Spiky wrinkly dude.
Spiky wrinkly dude. No, we've had suggestions of coffin fish.
Oh.
Yeah.
Okay. Should we try to get head on to see both eyes in the middle? I'll zoom out. We'll give it a go. He seems like he's pretty chill.
Got an hour to come up from there.
Trying to get that perfect shot.
Are we just going to come around a bit?
Yeah.
Nice.
So, yeah. Get both eyes straight.
I'm going to Google.
What do people say?
Coffin fish not a bad shot.
He seems happy just to let us photograph.
Oh, look at that.
So we're just trying to get the perfect shot.
Yeah, I think we've actually had a really good ID shout here from Nooney. Celeb's monkfish.
They eat monkfish. It's pretty good, isn't it? They eat that.
If he opens his mouth with that Nat Geo.
Alright, get in there.
I don't think he likes being photographed.
Look at that face that only a mother could love.
Yeah, I think so.
Yeah, hold that. Pull out with those teeth.
So the spikes are bifurcated. And
Okay, I'm happy. Thank you.
Thank you.
Thank you.
Right, then I'll call that.
Goodbye.
It's an excellent way to finish this dive. So good.
Okay, we say goodbye to this grumpy fish.
Yeah. Happy.
Happy. Pretty early. So today we saw awesome fish, collected awesome corals, saw a mysterious graveyard of corals, and we also did something with rocks. Is that right? We got some.
The geologists are super happy. But yeah, tomorrow we'll be at the same site, but we'll be skipping up a little shallower, starting at about 600 tomorrow. So yeah, we'll be in for another really cool dive. We'll be going up through the mesophotic zone, and yeah, it should be great. So hopefully see you guys all then, and say goodbye to this guy.
ROV bridge, so you think it's about 45 minutes, 50 minutes till it floats on surface?
This is math real fast.
Yeah, 20 meters a minute.
Yeah.
Right?
Yeah, sounds about right.
Okay, perfect, thanks.
Nice.
Shall we?
Ah, a fish, yes.